Hello, everyone. Good morning, good afternoon, good evening, wherever you are. Thank you very much for joining the IUIS EFIS International Day of Immunology celebration. We've got a really, really good lineup of speakers. My name is Faith Ossier. I'm the IUIS president, and I'll be co-chairing our session today. I'd really like to welcome our invited speakers. Thank you so much for taking the time to be with us. I'd like to welcome our participants to the roundtable. Again, we appreciate your time. Thank you for joining us. I'd like to thank the organizers, all the hard work that you've put in. I think we're ready to roll, so thank you so much. And I want to just welcome everyone from around the world. When we last looked, we had 106 participants, 106 countries represented. And so we want to welcome those of you who are joining on Zoom. We want to welcome those of you who are joining on YouTube and other streaming platforms. Really looking forward to what we've got before us today. And thank you again for joining. So my co-chair is the president of EFIS, the European Federation of Immunological Societies, Professor Salusto. And I'm just going to hand over to her now to give us some housekeeping, and off we go. Thank you.
Hello everybody. Also on behalf of the European Federation of Immunological Societies, I'd like to welcome you to the webinar to celebrate the Day of Immunology 2022. As you know, we started the webinar last year because of the current pandemic, and it was a great success, and therefore we decided to continue the tradition of having the webinar also this year. And the focus for this year is again on COVID and especially for vaccines. So we are very pleased to have a fantastic lineup of speakers and an interesting panel discussion after the presentations. And I hope you will enjoy the day and have some interesting feedback from the Day of Immunology. Just some information, please use the chat just to send us your comments, to tell us where you are, to ways, suggestions how to improve the webinar, whether you like it or not. And instead, you can also have pose questions to the speakers after their presentation. Please, however, use the Q&A function to have your questions. So probably we will not be able to answer all your questions or to have the speakers addressing all your questions, but at least we will send to them, and so they can probably give you some feedback through other means. So our schedule is quite tight, so I'm very pleased to kick off the webinar with, as you can see, the speakers that have kindly accepted to present. And we will start with Dr. Seth Berkley, that is the CEO of Gavi, the Vaccine Alliance. And Seth is really a pioneer in global health, public health, and is really a champion of equitable access to vaccines and ways to improve the access of vaccination and in general for to combat infectious diseases. So it's a great pleasure to have Seth joining us, and he will talk about the role of vaccines in the current pandemic situation. So thank you very much, Seth, for joining us today, and we look forward to your presentation.
Dr. Osia, Dr. Salusto, and distinguished members of the immunologic societies, thank you for inviting me here today on the International Day of Immunology to talk about the COVID-19 vaccine landscape, which has been and continues to be central to the entire global pandemic response. Right from the beginning, even before they were developed or before we knew that they would succeed, it was clear that vaccines represented our best potential defense against this virus. We worked with our partner CEPI to have the largest portfolio of vaccines across many different classes, viral vectors, adjuvanted proteins, inactivated vaccines, as well as the novel messenger RNA vaccines. Now, I don't need to tell all of you, but the normal rate of success for vaccines is low. And coming from somebody who worked for many years on HIV vaccines, for some agents it's been very difficult. Therefore, the expectation was that we might get one or maybe two if we were lucky, and that the timelines would be long, as the previous best timeline for a new vaccine was for mumps at around 4 years. But thanks to the incredible work of the scientific community, vaccine manufacturers, we ended up with more than 24 safe and effective COVID-19 vaccines, and in record time, with the first one taking just 327 days to develop and have regulatory approval. That is simply remarkable and obviously sets a new metric for us. As you know, CEPI has been talking about moving to 100-day timelines, and we just keep increasing those challenges, which is critical in a fast-moving pandemic. It's also remarkable that all the classes of vaccines worked, which is why we have so many vaccines available. But of course, we don't just need vaccines. We need vaccinations, and on a scale that had never been seen before. And not just for the wealthiest nations. Thanks to the largest and most complex global deployment of vaccines ever, led by a new organization or effort called COVAX, people in lower income countries were receiving their immunizations just 39 days after they first became available in high income countries. Now today, with nearly 11.5 billion doses administered worldwide, the COVID-19 vaccine effort has been one of the most incredible achievements in the history of immunology and vaccinology, and something we should all be proud of. And yet, as life begins to return to normal, particularly for people living in countries with high coverage, there are still more than 4.5 million new COVID infections each week and over 15,000 deaths. We need to realize that we are still in a state of global crisis. Given the unwavering ability of this virus to mutate, with a new variant emerging on average every 4 to 5 months so far, we have no reason to believe that this pattern won't continue. The threat of resurgences and potentially more dangerous variants still hangs over us. And even though we continue to hope for the best, that perhaps this is the last one and the epidemic is over, we must still prepare for the worst. So with so many shots of vaccine administered, why do we still have to worry about this? Is it because we don't yet have a universal or pan-corona vaccine one that reliably protects against viral transmission? Certainly in an ideal world, these could have potentially helped us stop the virus in its tracks. And obviously, we're hoping that those will be developed going forward. But in fact, even as this coronavirus has continued to mutate, the vaccines we have remain highly effective at protecting people against severe disease, hospitalization, and death. So I would argue that we have what we need to mostly finish the job. The reason COVID remains a threat globally is for the simple reason that the global access to vaccines has not been equitable, and because the virus continues to have ample room to circulate and mutate. Today, 2.7 billion people have still not been vaccinated. The vast majority of these are in the world's poorest countries, but of course we're seeing with vaccine hesitancy many in high-income countries still avoiding vaccination. Now, most wealthy nations have coverage around 80% or above, and they're now offering fourth booster shots. Some lower-income countries, 18 at the last count, have still yet to reach 10% of their population with 2 doses. The good news is that we can likely end the severe effects of this pandemic with the vaccines we have, provided that coverage is high enough, including boosters for high-risk populations, in all corners of the world. We just need to make sure that all countries are able to achieve their national vaccine targets. This today is our number one challenge. It is also why, even in the face of other pressing global crises, like in Ukraine and Afghanistan, we must remain vigilant and ensure that COVID remains a global priority. Despite immense challenges, COVAX has made and continues to make great progress in helping countries increase their coverage by making access to these vaccines more equitable. To date, we have shipped more than 1.4 billion doses, with the vast majority, nearly 90%, going to lower-income countries through the Gavi COVAX Advanced Market Commitment, or AMC. Without the COVAX AMC, people in these 92 countries, which collectively make up roughly half the world's population, would have struggled to get access to these vaccines. And with COVAX's support, on average, 45% of people in these countries are now protected with 2 doses. As the only truly global multilateral solution, we couldn't have done it without the incredible support of the international community. However, we are not done yet. Populations in AMC countries where health systems are stretched and often have to deal with multiple public health problems can be hard to reach. Thanks to COVAX delivery support, the number of countries with coverage of less than 10% may have dropped from 34 in January to just 18 in the last 3 months, but delivery remains a great challenge. Thanks to the generosity and ongoing support of G7 and G20 nations, not least the Italian government, whose leadership during its G20 presidency last year was critical, as well as the European Union, the African Union, CSOs, and other partner organizations, earlier this month at the Gavi COVAX AMC Summit, we raised the cash and innovative financing required to provide these countries with additional delivery support. This will help get more doses into more people sooner. Aside from deliveries, other uncertainties remain, not the least the potential for fresh resurgences or new variants. We are currently getting the doses we need, but we need it to stay that way. In the coming months, there is a risk that we could face further constraints to vaccine supply, the need for additional coverage of particular vaccines, or perhaps the need for new variant-specific vaccines. In all cases, we need to keep these doses flowing, not just to wealthy countries, but to developing countries. That is why at the Gavi COVAX AMC Summit, we launched the Pandemic Vaccine Pool to protect COVAX's supply from future disruptions. In a sense, this insurance policy is the first test of the international community's commitment to pandemic preparedness, because it will ensure that we are prepared for whatever else this pandemic and this virus throws at us. That is why the pandemic vaccine pool is so important and why it is so encouraging to see such widespread support for it at the AMC Summit. And I look forward to working with governments, including Italy, to ensure that it is fully funded and a success. Ultimately, it is only by working together through international collaboration and partnership with the best science that the world has to offer that we can end COVID. We are only safe if we are all safe. Thank you for listening.
Thank you very much, Dr. Berkley. That was really fantastic and a really impressive effort to get vaccines everywhere. I just have a couple of questions that are to pose to you. And the first is really about the pandemic vaccine pool. So you've elaborated that this will prevent further disruptions during COVID. But if we think about the context of future pandemic preparedness, how do we ensure that the next time there aren't disruptions in the first place?
Well, with COVID, the main challenge to global supply had started out being vaccine hoarding by wealthy nations, export restrictions on vaccines, as well as the material and components used to make them, and delays in scaling up the manufacturing, given limited manufacturing capacity around the world. The problem we face is that in a time of crisis, you cannot assume countries or companies won't put the interest of their own citizens or shareholders first. Obviously, in some sense, it's their duty to do so. So the best and most sustainable way to mitigate against this is to increase global production capacity and create greater geographical diversity in that capacity. That means investment in the development of regional manufacturing sites, especially in Africa, where they're less present. This in itself, of course, is not without its challenges. For example, who's going to pay for it? How do we ensure that these facilities don't sit idle between pandemics? But if we succeed, it could ensure that more people across the world are protected more quickly, and that's in everyone's best interest.
Absolutely. Thank you. And just a bit more on the pandemic pool. You said that this is the first test of pandemic preparedness. What would you say we need to do now to prepare for the next pandemic?
Well, the first thing that's critical is to recognize and truly understand that there will be a next one. It is an evolutionarily certainty, and novel viruses will emerge, especially with relentless deforestation, and global trends are making it much more likely that such outbreaks will escalate into pandemics. In dense population and growth, urbanization, rapid global travel, and climate change all will hasten the spread of a virus. So even after COVID, infectious diseases will continue to be a threat to global health security. We also need to learn from COVID and not wait until the next pandemic strikes, which means taking action and putting funding in place even before you know what you're up against. Excuse me. If you wait until the crisis strikes, you lose valuable time in setting things up and raising funds. That certainly delayed things with COVAX, as we had to get staff and raise finance before we could go ahead and purchase vaccines. We also need to recognize the value of what we already have in place for pandemic preparedness and strengthen it. By increasing routine immunization, organizations like Gavi are preventing epidemics and pandemics on an ongoing basis by protecting people from deadly infectious diseases. At the same time, these immunization programs help to make stronger and more resilient primary health care systems, which besides supporting people with their health care needs, also serves as a vital part of the early warning system for emerging infectious diseases. We need to ensure that these kind of ongoing efforts get all the support they need and that we pay attention to the Sustainable Development Goals kind of mantra, which is leave no one behind.
Thank you. Listening to you, there are really many moving parts. What could you crystallize what you think the single most important part of pandemic preparedness is? What's the one thing that we should think about?
Well, Faith, that's a really tough question because what we've seen related to pandemic is that there is no one single solution. Vaccines, for example, are, as far as I'm concerned, have been essential, but we don't just need vaccines. We need vaccinations. We need all the pieces necessary to turn these into shots, including delivery systems, cold chains, syringes, et cetera, healthcare workers. A thing to remember, and I already touched upon it, is that much of what we need to fight the pandemic was already in place, the infrastructure, resources, and expertise. But it's only by working together that we really have been so effective at scaling up so quickly. So what I would say is continued global collaboration is probably the most essential part of pandemic preparedness and response. It's only by coming together and building on the existing global health networks, leveraging existing infrastructure, resources, and expertise that the global community can ultimately beat a pandemic. Finally, we need to value science and truth. The politicization of the public health response and dishonesty and lack of truth has led to real challenges given social media and the internet has moved around the world at the speed of light. And that's why your community coming together like this and sharing experiences globally is part of the solution. It's an immunization against all of that misinformation and lack of truth. And so my hat is off to you for all that you're doing to try to create a global community to be thinking about the important work of immunology and what needs to happen.
Thank you very much. So Dr. Berkley, we really, really appreciate your time and your insights and really important that we are all in this together. and that none of us is safe until all of us are safe, and we must continue to collaborate. So we just really want to appreciate your time. We know your schedule is really tight. On behalf of the IUIS, EFIS, everyone online through all the different streaming platforms and on Zoom, we just want to say thank you, Dr. Berkley, and we really appreciate you. Thank you very much.
Thank you for having me, and very good luck with these important proceedings.
Thank you. So it's my pleasure now to introduce our next speaker. Really happy to introduce Professor Antonio Bertoletti. He trained as a specialist in infectious diseases at Scripps and the University of Parma in Italy. He was the first to characterize hepatitis B virus-specific CD8 T+ cells, and in 2006, he became the director of the Infection and Immunity Program at A-STAR's Singapore Institute for Clinical Sciences, and then joined Duke NUS Medical School in 2013. So he's going to speak to us about Act Early and At the Right Place, kinetic and localization of SARS-CoV-2 specific T cells after infection and vaccination. Welcome, Antonio, and thank you for accepting our invitation.
Thank you. Good morning, good afternoon, and good evening to everybody. My name is Antonio Bertoletti. I'm working at the Program of Emerging Infectious Diseases at Duke NUS Medical School in Singapore. Let me thank first the organizer of this International Day of Immunology to invite me to speak about some of our work on SARS-CoV-2 specific T cells. In particular, today I'm going to present some work where we try to show you that actually the rapid deployment of virus-specific T cell response is very important for viral control with limited pathological consequences. And then I'm going to show you new work, still unpublished, about the presence of tissue-resident SARS-CoV-2 T cells in vaccinated and in subjects that are being infected after vaccination. So, but let me go first to the work where we analyze the dynamic changes of virological and immunological parameters that were present in subjects that were SARS-CoV-2 infected, and they have actually or severe or mild disease. We studied very few subjects, I have to say, but we studied them at several time points during the acute phase of infection, when there was still high level of viral replication. Different time points where we follow them until some of the patients control the infection without symptoms or with very mild symptoms. And then we study the subject during the convalescence. And what we have done was to quantify initially the quantity of virus. We studied then the antibody response with different methods. We studied the quantity of neutralizing antibodies and also the quantity of antibodies specific for the whole spike, for RBD, and for MP. We also then studied SARS-CoV-2 specific T cell response. The response was analyzed initially with simple ELISpot assay, but we test the T cell response against different structural and non-structural proteins of the virus, utilizing a pool of peptides, 15 MERS, overlapping of 10, that were covering these individual proteins, MP, membrane, spike, and other different non-structural or accessory molecule of SARS-CoV-2. So what we found? Well, again, initially, and I want to draw your attention on the fact that I indicated with green the patients that have mild COVID-19, whereas the patients indicated in red are the patients that have severe COVID-19. And what we find at the beginning was clearly that the quantity of virus and the duration of virus was directly proportional to the severity of the disease, something that other groups of them demonstrated that the quantity of virus was really directly proportional to the severity of the disease. And then we look at the production of antibodies. Here, again, you can see red lines are the subject with severe COVID-19, green lines are the subject with very mild COVID-19. And you can see that the antibodies were produced in both groups, but the patients with severe disease are actually quite high now, quantity of antibodies that were actually reaching a level that were superior to the patients that have mild COVID. And similar data were not present by measuring only neutralizing antibody that are indicated in this initial picture, but also for antibodies specific for RBD, for the S1 region of the spike, and also for nucleoprotein. The picture was completely different instead when we were analyzing SARS-CoV-2 specific T cells. Here I indicated the quantity of spots that were generated summing up all the responses to all the different structural and non-structural proteins. So basically, this is the quantity of the global SARS-CoV-2 specific T cell response that was induced in patients with mild, moderate disease or in patients with severe disease. And you can see it by yourself that there was a rapid induction and a very strong response in the subjects that have mild, moderate disease, something that we could not see in patients that have severe disease. And actually, in the patients that unfortunately have a fatal disease and die from SARS-CoV-2 infection, we were not able to detect any antigen-specific T cells. So again, this data somehow demonstrated a high frequency of SARS-CoV-2 specific T cells was associated with mild disease. Not only the high frequency, but also the time of the appearance was really correlated with a really shorter duration of infection and mild disease. Here, we put basically the day that were, let's say, the first detection of antigen-specific T cell response and the day of the clearance of the virus. That correlate perfectly. But similar data were not, let's say, similar correlation was not present instead for antibodies. So therefore, again, this first part of the talk, I present this data that have already been published that show, despite, let's say, a small number of subjects that we have analyzed, show clearly that there is the early induction of T cell response, which were in the patients also linked with antibody, however, but however very important for the rapid control of SARS-CoV-2 infection. Now, these data were actually supported by much, let's say, other studies that have been published, for example, recently, and studies that were done in much larger population. And again, this work, for example, done in UK, has further supported the importance of this rapid induction of virus-specific T cells. Here, the researchers, they were studying subjects that have asymptomatic infection, and they were able to demonstrate that these subjects are all characterized by a rapid induction of interferon alpha that was immediately followed by the production of virus-specific T cells. Okay, now I hope they say this data convince you that it's important actually to have a T cell response that it should act really fast. But I will say it's probably also important that it should act in the right place. And the point is that SARS-CoV-2 is not infecting the blood, but is actually infecting the upper and the lower respiratory system. And in particular, there have been data that have shown that nasal ciliate cells are actually the cells that are first infected by SARS-CoV-2. And importantly, in data in vitro, I've actually shown that these cells are able to maintain very high level of viral replication, despite the fact that these cells are actually producing interferon alpha and activated innate immune response within the cell. So the SARS-CoV-2 seems really to be able to replicate in these cells, despite activation of innate immunity, at least in vitro. And this again shows that it could be really important to have cellular immunity that could destroy these infected cells. Do we know whether T cells, SARS-CoV-2 specific, are present in the nasal cavity or, let's say, in the upper respiratory system? Well, there have been already data. In particular, there have been a work done in Karolinska by the group of Markus Burger that have demonstrated that there were T cells specific for SARS-CoV-2 also in subjects in the oral cavity, in the lymph node tissue of the oral cavity of healthy individuals. And these were probably cells that were cross-reactive, they were induced by seasonal coronavirus. And then we have a nice work that was published, was done by the work done at the University of Leiden, where they demonstrated that infection in the infected people, we have clearly recruitment of T cells in the nasal cavity. And they were also able to demonstrate by sequencing the TCR, the spike-specific T cell could be detected in the nasal cavity of the infected subject. In addition, I mean, we have many work in mouse model that are demonstrating that the induction and the presence of memory T cells, CD4 and CD8, is important for the control of viral infection in the animal model of SARS-CoV-2 infection or in the animal model of coronavirus infection. This work, for example, is a seminal work of Professor Perman that demonstrates this already, I would say, 4 years before the pandemic. Now, the question that we want to answer in our work was really to understand first whether in subjects that were only vaccinated, so subjects that have 3 doses of messenger RNA, mainly BioNTech Pfizer vaccination. I mean, this vaccination could actually induce some level of mucosal T-cell response, spike specific, or whether, let's say, this SARS-CoV-2 specific T-cell can be induced only by the infection. And then we were interested to understand whether the presence of spike-specific T cells that we know are induced in vaccinated subjects, at least in the circulatory system, can actually bias the repertoire of the SARS-CoV-2 specific T cells after infection. And so the first point was really to try to have these nasal T cells. And we studied them in 14 subjects that were vaccinated only, and in 15 subjects that were vaccinated and then have an infection, probably an Omicron infection, since these were subjects that were studied in Singapore, and 95% of the virus that was sequenced here in the last 4 or 5 months was really an Omicron SARS-CoV-2. Then we have done the phenotype, simple phenotype, really looking whether in the nasal secretion we can detect T cells, whether they were CD4 or CD8, and whether they were expressing tissue resident markers. And then we analyzed SARS-CoV-2 specificity with 2 different methods, with ELISpot or with flow cytometry, with a modified, let's say, activation markers analysis that I will explain later. So the first point was really to understand whether we could collect a sufficient number of T cells from the secretion that we collect from the swab. One point is that we didn't use any sort of scrapping, but we used, let's say, the normal swab that is used for antigenic test. And this swab was basically used to collect the secretion. And then the spot was put in RPMI and we tried to liberate the cells. And to our surprise, we obtained quite a large number of lymphocytes. Here, I'm indicated the, let's say, appearance of the cells that were collected from the nose. And you can see that there were clearly a population of lymphocytes. There were CD3 and there were mainly CD8. And the number that we obtained was actually substantial. We could obtain 70,000 cells in this individual. One point, the patients that were infected, so they were infected after vaccination, were in any case studied at least 3, 4, 5 weeks after infection. So you see here, therefore, we are not looking at any acute infection. We are looking more at a sort of memory immune response induced by the infection. And the phenotype of the cells that we collect from the nasal was clearly different from what we can see in the peripheral blood. They were really mainly expressing resident marker. They were CD103 positive and CD69 positive. And here you can see it clearly. The cells were mainly CD8 cells from the nasal. They were about 50–60% of the T cells that were collected from the nasal were CD8 cells in almost all the individuals that we studied. And the CD8 cells were really 90% of them were really tissue resident T cells. CD4 T cells instead, no. CD4 T cells, they were, I would say, they were different frequency, but in the majority of the case, the cells were not CD103 and CD69 positive, but were only CD69 positive cells. And now, then we study the SARS-CoV-2 T cell specificity. And to do that, initially, we have done an ex vivo direct ELISpot assay, where we were very careful to at least analyze 8,000, 10,000 cells per well. And each well was stimulated with individual pool of peptides that were covering individual proteins. And in particular, we studied the NP, response against NP, against membrane, against spike, and also against NSP12. NSP12 is the polymerase of SARS-CoV-2, and we have demonstrated with work done in collaboration with Malamaini at the University College of London that a response to NSP12 seems to be extremely important for the protection, at least in subjects that have an abortive infection. And the other important points that I want to point out is that we consider an ELISpot assay only if we were able to detect at least 100 spots in our positive control. So we were very careful to have, I will say, an ELISpot assay done with a number of cells that can tell us something. And here are the results. And the results were clearly, I would say, black and white, because this is the results of the vaccinate-only subject, nasal secretion. And you can see it by yourself, despite the fact we were able to find a very good positive control, we were not able to see a single SARS-CoV-2 specific response. Such a response was instead present in 14 out of the 15 subjects that were vaccinated and then infected. And you can see by yourself that the response was not only specific for spike, which is indicated with the red color, but was also specific for other SARS-CoV-2 antigen, NSP12, NP, and membrane. And importantly, and this is something that I'm showing here, I would say most of the individuals were actually able to mount a T-cell response able to recognize different proteins. You can see that most of the subjects recognize at least 2 or 3 pools and even 4 pools of peptides that were covering different proteins. Therefore, the T-cells of the nose, they really don't recognize only spike, but have a sort of much broader T-cell response able to recognize different SARS-CoV-2 antigen. And this was detected only in subjects that were vaccinated, but then have an infection. What was the hierarchy? What was the difference between, let's say, SARS-CoV-2 specific T cells in the blood and in the nasal cavity? So here I'm showing you this data. The first point that I want to make is that the frequency of SARS-CoV-2 specific T cells appears much higher in the nose than in the blood, at least in the 3 subjects that at the moment we have done. This is still a work in progress. In one subject, instead, we find that the frequency was higher in the blood. But the other point is that the hierarchy of the recognition of the different viral proteins is actually, I would say, different. In some subjects, we have similar hierarchy. Spike-specific T cells in this subject was dominant, both in the nasal cavity and in the blood. But in other subjects, you can see that the response in the nasal cavity was completely independent of what we detect in the blood. And this is, for example, the case of this subject. We has a classical and very strong NSP12 response in the nose that was barely present actually in the blood. Whereas in the blood, you have a dominant spike-specific T-cell response. Again, this data seems to indicate that what is induced in the nose is not directly correlated with what we can find in the peripheral blood. And then we were trying also to understand whether these spots, these T cells producing gamma, they were actually CD8 and CD4. We ran on some difficulties, technical difficulties, because the number of T cells is extremely low. But again, then we designed these experiments where basically we have purified monocytes from the blood of the subject. We pass them with the different mixture of peptides, and then we had the nasal cells for a very short times. And then with a flow cytometry, we analyze the expression on CD8 positive or CD4 positive of CD4 T ligand or the degranulation marker CD107. It was basically a modified activation marker assay. And what we were able to find, we were able to demonstrate that there are CD8 T cells in the nasal cavity that can recognize not only spike, but also nucleoprotein, and in this case, for example, NSP12. So we have clearly induction of CD8 T cells able to recognize different SARS-CoV-2 antigen. For CD4 T cells, we also find that there were, in some subjects, CD4 cells, but these CD4 cells, they seem to be specific only for spike. Whether these CD4 cells are actually cells that were initially primed by the vaccine and then are recruited in the nose is something that at the moment we don't know, and is something that we are studying. Finally, how long this nasal CD4 or CD8 T cell persists. We have studied them now in 3 subjects for at least 60 days. And we can see that at the moment, the frequency is pretty much stable. Here, for example, you can see the subject have initially a response to the nucleoprotein, to spike, and to membrane. The membrane seems actually to go down in a couple of time points, but the 2 response to spike and MP are persisting for 60 days, and the same for this subject that clearly have a dominant NSP12 response. So far, at least, it seems that the T cells, SARS-CoV-2 specific, CD8 and probably CD4, are able to persist for at least 60 days. So in conclusion, what I show you here is that SARS-CoV-2, probably here an Omicron infection, in vaccinated individual induce a localized SARS-CoV-2 specific T cell response. And this response was clearly not detected in subjects that were vaccinated only. The other point that I think is important is that the vaccine-induced spike-specific T cells, all the subjects were vaccinated and they do have spike-specific T cells in their circulation, do not suppress at all the induction of T cells specific for other proteins. We were able to demonstrate presence of NSP12 or MP specific CD8 T cells in the nasal cavity of subjects that were vaccinated. Now, the presence of this cell might actually explain why there is a better ability, basically, of the subject that have this sort of hybrid immunity to control SARS-CoV-2 infection. And this is something that now, epidemiological studies are starting to really detect. I want finally to make another point. I was just to say that this, let's say, the profile of hybrid immunity, the profile of SARS-CoV-2 immunity that is present in subjects that have been vaccinated and then infected seems to be really peculiar. We have been already worked, for example, here I'm quoting the work of Marion Pepper that have shown, again, in subjects with hybrid immunity, that there is a much broader B-cell response repertoire, and the CD4 T cells here are of a peculiar functional profile. They are producing gamma, like exactly the nasal cell that we found, but they are also producing some level of IL-10, which might be important actually for, let's say, containing inflammatory event. And this is something that also we find the similar profile in subjects that have a symptomatic infection. And also, again, subject with hybrid immunity, this is the work done recently by the group of Paul Thomas. Again, it shows that even if you're vaccinated, you can actually induce a much broader and differentiated CD8 T cell response. Again, showing that the initial vaccination is not reducing your T cell responses against SARS-CoV-2. And now we are also showing in the subject with hybrid immunity that there is induction of nasal T cells, of SARS-CoV-2 cells resident in the mucosa that are probably important for the control of viral infection. So thank you very much for your attention. I hope you will have a question. Here is the photo of my group. I want to thank them all. There is no time, but I was also to really thank all of the clinical collaborators that they give us a lot of fantastic samples. Thank you very much for your attention, and have a good International Happy Day of Immunology. Bye-bye.
Thank you very much, Antonio. That was a terrific talk, very inspiring, a lot of data, and congratulations on taking over such a complex study of analyzing T cells in the nasal cavity. We know how much it's difficult to perform these experiments. So we have some minutes for questions, and I see that there are many questions in the Q&A chat, and so I'll try to pick up some of them, but please, if you have time, Antonio, answer to some of them after this session. Thanks for the nice presentation. Emanuela Corsini says, while Omicron seems to affect the upper respiratory tract, what about the presence of nasal CD8 cells with other COVID-19 variants?
Well, I mean, to be completely honest, I don't think there would be any difference because in any case, I mean, also the other, let's say, SARS-CoV-2 variants were infecting upper respiratory tract. The fact is that clearly, again, I don't know what it could have happened before. Honestly, I also stress the fact that, I mean, I suppose that the infection of the subject that we were testing was Omicron, even though I'm not having any real demonstration. But to be completely honest, I mean, I do think, I mean, what we're showing is that if there is an infection in the upper respiratory tract, there could be T cells that or are recruited from, let's say, local lymph nodes or are, let's say, directly induced in the nasal cavity, and they seem to persist at least for 2-3 months. I can say now that actually they can persist for 3 months.
Yeah, that's really good. Yeah. So actually, we have also an interesting question, more general question from Adelaide Surakan. So many data pointing to the potential value of intranasal boosters, yet it does not seem to be a priority among vaccine developers, producers. What is your opinion about
I don't know. I mean, yes, certainly so far, let's say there is not yet a vaccine that is usable in subject, but to be honest, I think the studies about nasal vaccine are really progressing. And I think, you know, Akiko, for example, have shown recently that it's possible to induce, at least in animal model, the T cells only using a protein with adjuvants. So again, animals that were initially vaccinated and then with an adjuvant carrying a single proteins, they were seeing some induction of and presence of T cell in the nasal cavity. I do think, to be honest, even though again, I'm not working on vaccine directly, but I'm just looking at patients that have been vaccinated and understand what's happening, that there is now a growing interest. And again, clearly, again, I'm studying only T cells, but I know also there is now an interest on looking at IgA, of production of antibodies also in the local nasal cavity. So I do think there is now this interest is growing, and I suppose, again, that we can see something that could be available soon. I know also that in a way it is somehow difficult to induce a good mucosal immunity, and that was something from studies in influenza. But this is not exactly my expertise.
But you're right, that's an important point. So Branka Gruber-Bauch is thanking you for the great talk. It was, if one was to harness the potential of mucosal tissue-resident memory cells for COVID vaccination, how do we avoid the tissue injury due to their cytotoxic action?
You know, I mean, it's always a little bit the sort of problem of T cells that, of course, they have also to lyse some of the infected cells. But again, I more an expert, I would say, of hepatitis B virus than on SARS-CoV-2. And also in hepatitis B virus, which is a non-cytopathic virus, we can see that the T cells, even though they are inducing some level of inflammation, are essential for the control. I'm really thinking that, you know, the real damage, and in a way, we can see it also in SARS-CoV-2 infection, the COVID-19 severe seems to be more due to a sort of uncoordinated immunological events with, let's say, low level of antigen-specific T cell induction. And this is also something that I show at the beginning of my talk, where in reality, we have seen that, at least again in the peripheral blood, the patients that were able to mount an immediately a T cell response were the ones that were able to control. So yes, I mean, there could be clearly some level of inflammation and there could be some like a small side effects, I mean, some problem of damage of a mucosa, but you know, the importance of the T cells is really to eradicate the fonts of viral replication. So I might be biased on this view, but I do think most of our data are really pointing that the T cells are more protective when they are in a good number and they are functionally able to produce cytokines and lyse.
So actually, I wanted to ask also something about the initial data you showed about severe and severe COVID and less severe, and the fact that you didn't find T cells in the severe cases, and I wonder whether it's a question of localization, and in a way also going to the question that that Sunit Singh is asking whether there is exhaustion in COVID-specific T cells, especially probably in patients with the severe cases.
Yeah. Okay, so I would say that from my data, you are right. We only study blood, and from the blood, we can only see this difference. So one possibility is, okay, when they have severity, great severity of a disease, all the T cells are present in the lung. However, here I have to quote other work of different groups that, again, in reality, also looking at T cells in the lung. In the case of severe disease, most of the T cells which were present, some of the T cells were present, but in reality they were non-antigen specific. And again, instead of the contrary, in subjects that have mild disease, they were find antigen specific T cells. So again, clearly my data were not, let's say, able to disentangle this. Exhaustion, again, unfortunately, I don't have the data to answer whether the lack of, let's say, ELISpot response was due to exhaustion. And again, I have here to quote other paper. There is a certain, I would say, lack of data during the acute phase of the disease. In general, in SARS-CoV-2, there are not many data. However, if, again, I'm looking at what's the study that have been done by several groups on tetramer positive cells, they will say that some people are really sure that the cells were PD-1 positive, but they were still functional. So they were activated and they were not classically exhausted cells. So I would say that perhaps we can say that the jury is still out, but as far as I can see, I don't think exhaustion is playing a great role. It's more an uncoordinated activation of T cells, B cells, that then they're causing inflammatory events.
Yeah, that's a good points, and certainly still some open questions in the field that need to be answered. But thank you so much, Antonio, for this wonderful lecture.
Thank you very much. Thank you for the question.
And as I said, you have certainly more questions in the Q&A section, and they would be great if you can answer to the participants that I see are growing. We have many people joining, and please use the chat to tell us where you are and whether you have comments and suggestions on the webinar. So thank you again, Antonio.
Thank you, everybody. Bye-bye.
And now it's really my great pleasure to introduce the next speaker, Rita Carsetti. Rita is the head of the B-cell pathophysiology research unit at the Bambin Gesù Children's Hospital in Rome. In the last year, she has been involved in several projects related to the development of the immune system in children and adults, and the change due to different types of immunodeficiency. She's also involved in increasing the knowledge and public awareness on vaccines and vaccination, and collaborates with patient association. She's a founding member of the European B-cell Network and also a very active member of the UIS, and in particular, she's the chair of the publication committee within the UIS. So she's really a very well-known and active member of the immunology community, and so we are very pleased that she has time to join us to talk about the role of memory B cells in COVID infection and vaccination. Thank you, Rita, for joining.
Thank you, Federica. Thank you very much for inviting me today to give a talk on memory B cells in COVID-19 infection and vaccination. I share my screen. So when we have an infection starting at mucosal site as the most frequent infection, we have a lot of defense system already in place if the pathogen is already known to us. For example, let's imagine that this is an influenza virus. We will already have secretory IgA post on the surface of epithelial cells working as a primary system of defense, a very early system of defense. If the virus can cross the epithelial cells, then we will have antibodies under the mucosal level, at the mucosal level, and we will have resident memory T cells and memory B cells. And memory T cells will immediately kill the infected cells, and memory B cells will produce tons of antibodies when they're mostly needed to stop the virus. Then we also have an additional system of defense, which is the adaptive immune response occurring in the germinal center. But this is it has a longer kinetics. In fact, this was the problem with COVID-19, because most of us didn't have, actually nobody had preformed antibodies, because this was a new virus, so we didn't have IgA. Some of us had some cross-reactive antibody from previous infections, but mostly we defended ourselves with the innate immune system. The response in the germinal center, of course, late. There are 2 weeks of time needed. In fact, when we have the primary follicle, here the B cells start to react together with T cells, the work is quite long, because B cells have to proliferate in the germinal centers, they have to introduce mutation in their immunoglobulin genes, they have to select for antigen specificity the best antibody, and only when this has all happened, we will have memory cells, short-lived plasmablasts, and tons of antibodies in our blood, which will clear the infection. Some of these plasmablasts, which are called short-lived because they live for a couple of weeks, will become long-lived plasma cells, and long-lived plasma cells will go to the bone marrow and remain there for a long time, where they inhabit a special niche, a dedicated niche. Memory plasma cells produce only low amounts of antibodies, but this is not so bad, because I always have my memory cells that in case of reinfection will immediately produce the high amount of antibodies. So the long-lived plasma cells make the antibody that protects me. Meanwhile, I make enough antibody from the memory cells. If the infection was at the mucosal side, then some of the memory cells will have migrated there and will have become resident memory cells. So they will protect also the mucosal side. During COVID-19, there was a real problem with the germinal center, because the severe disease with all the inflammatory cytokine disrupted the germinal center. So the adaptive immune response did not occur at all. It occurred, however, in patients with less severe disease who didn't die from COVID-19. And this fantastic work from the lab of Michel Nussensweid first showed that you could measure with fluorescent antigens memory-specific cells, and you could also sequence these memory cells and see that they had somatic mutations, so they had been generated in the germinal centers. The nice discovery was that 6 months after the infection, the memory cells were still there. Not only they were still there, they had increased in number, but especially they had more mutations, meaning that the persistence of antigens in our body allows the cells to improve progressively their specificity. So B cells couldn't play a fundamental role in the course of the primary infection because they came too late. But they can play a very important role in the prevention of disease because they can produce neutralizing antibodies, thus blocking the interaction between spike and the ACE2 receptor. How do we do these memory cells and this very fantastic neutralizing antibody? We can produce them with vaccines. Vaccines works because they induce immunological memory without causing the disease. So we could do the germinal center reaction, we select the cells, and we are left with the memory cells which are specific for COVID-19, for SARS-CoV-2, and they are of IgM or switch isotype. The switch memory cells normally have more mutation than the IgM memory cells, and we also have long-lived plasma cells that have been demonstrated in COVID-19. Immunological memory is the proof that B cells are functional, and you have to think here about all the people who are not able to generate memory cells as people with primary and secondary immune deficiency. And their failure to generate memory cells can explain what is called vaccine failure. So we studied memory B cell development and performance in a group of healthcare workers of the hospital where I work. And we also tested where they were functional, studying people who had breakthrough infections. And what we found out was interesting, and I want to share with you. So, first of all, we confirmed that, as already many others have demonstrated, antibodies produced in response to the second dose of vaccine rapidly declines in the serum. They become very low already 3 months after the vaccination, but 9 months they're really low as compared to the immediately after the vaccination. This has scared lots of people, but it's very normal physiological immune response. After every vaccination, we have an immense amount of antibody produced by the short-lived plasmablast I've shown you before. Then those die. Then we have the long-lived, the memory plasma cells surviving. Then when we have a booster or we have an encounter with a pathogen, we produce again lots of antibodies. These antibodies will decline again, always because only the few long memory plasma cells that survive in the bone marrow will produce this standing amount of antibodies which we have. Do the memory B cells develop in vaccinated people? Our workers were vaccinated with an mRNA vaccine, and we developed a system to detect high affinity and low affinity memory B cells. Low affinity because we use fluorescent spike labeled with 2 different fluorochromes, one very bright and one not very bright. And you can see low affinity and high affinity memory cells. And you can also ask where are the RBD specific cells, and you see that they are only in the pool of the high affinity memory cells because they are being generated by the germinal center reaction. So you can see that before vaccination, everybody has some low affinity memory cells, which become high affinity 1 week after the second dose. The real surprising thing was that if we measure the level of the frequency of specific memory cells months after the second dose, you can see the memory cells increase in time. This was surprising, but this can be explained by persisting germinal centers where the antigen is still contained, recycling their memory cells improve the specificity as it has been demonstrated after the disease. And you can see that with time, low affinity memory B cell decline, whereas high affinity memory B cells increase, and of course the RBD specificity is maintained. So the memory B cell continue to increase even 9 months after the vaccination. Low affinity memory B cells are mostly of IgM type, high affinity memory B cells, as suspected, are mostly of IgG or IgA type. Thus, whereas antibody decline, our memory cells increase. So why are we so worried in reality? Do the memory cells work? Yeah. So we studied this by comparing the immune response of healthcare workers who did not have any infection to healthcare workers who had breakthrough infection, and where we measure, antibody and memory B cells levels at different times after the infection. First of all, our first question is, do I get an infection, a breakthrough infection, because of my waning immunity? No, there is no waning immunity, really. These are people which are very healthy and very young and are compared to their colleagues. And these people got infection 3 months after the second dose compared to people who were vaccinated 3 months before. These people instead got infection 6 months after the second dose, and these are also healthcare workers who didn't get any infection 6 months after the second dose. And you can see there is no difference in the amounts of neutralizing antibodies, and also there is no difference in the amount of spike-specific memory B cells in the peripheral blood. But do the memory cells work? Yes. And we demonstrate this because we see that the anti-spike, anti-RBD reality specific antibody increase at the 2 or 3 days after the infection to our, to a very high level. And antithromeric spike antibody also increased 2 to 3 days after the infection. That means I'm not doing new germinal centers because I'm very fast. In fact, anti-N antibody increased 15 days after the infection, because anti-N is not contained in the vaccine, so I cannot use memory B cells to make these antibodies. I really have to make a new germinal center. Spike-specific memory B cells also increased in frequency in the blood and increased quite fast at 2, 3 days, 4 days, 5 days, and so on. RBD-specific cells are there. And also we can measure the increase of specific plasmablasts, because plasmablasts still express an immunoglobulin often. So you can measure specific plasmablasts in the blood, and you see that they increase immediately after the positive swab. So, but why do I get the positive swab if my memory cells work so well, if I have been vaccinated? One of the problems is, probably the most important problem, is that the virus is invading not my blood, but my nose. And if I am looking at salivary IgA that are really increased in the blood of people who had COVID-19 even months before our measurement, you see that the IgA in the saliva is there. But salivary IgA in people who just got the vaccination is not so high. This is instead increasing and very rapidly in people who get a nasopharyngeal swab, positive nasopharyngeal swab, indicating that after vaccination, I generate lots of antibodies that may transudate to places of the tissues, but also to mucosal sites. And of course, this amount of antibody that transudate is proportional to the concentration of antibodies in my serum. So I will have more transudated antibody immediately after the vaccination and less later. In reality, what we do in response to a breakthrough infection is that we really send our memory cells where they need it, because that's what we really want, to make resident memory cells. And this does not occur after parenteral vaccination, because the memory cells need to be addressed to the places where they need it. So, for example, here mucosal vaccine will also help to generate not only systemic, as we already have, but also local defenses. So what we really learned is that when we study only antibodies, we only look at the tip of the iceberg that is our immune memory. There is a part of our immune memory that is under the water. And here we have memory B cells, memory T cells, whereas long-lived plasma cells are really producing only a small amount of antibodies, but we are not unprotected. We just don't have to see only the tip of the iceberg. An additional problem that is occurring right now is that the virus continuously mutates, because this is normal evolution. So we have the variants. When we see that monoclonal antibodies are not able to neutralize these viruses anymore, we have to choose new monoclonal antibody to treat people. But thanks to that, and because we're lucky, the human B-cell response is not monoclonal. We have demonstrated a few years ago that our memory pool is composed of two types of cells, CD27 dull and CD27 bright cells, and the dull cells have less mutation, the bright cells have more mutations. As you can see here, these are the dull cells, these are the bright cells, this is the frequency of mutations. And this means that that our memory is organized in large clones of cells which have a precursor with few mutations, and then lots of daughter cells with different amount of mutations that can thus adjust to, for example, variant of the virus. Plus, you have to consider that we don't produce only one clone against a certain antigen. Each one of us can produce antibody with different VH and different V light chains in response to the same antigen. These cells work very well because you can see here, this is a person seen 9 months after the second dose. This is 1 week after the third dose, and this is 1 week after this person got Omicron, the Omicron variants. So you see that the memory cell generated by the vaccine also recognize the Omicron variants and make lots of plasmablasts and antibodies when they are needed. 9 months of the second dose, this person wasn't producing plasmablasts because there was no ongoing SARS-CoV-2 specific reaction. This is 1 week after the third dose. Okay, also the plasmablast increase, and this is the same person after Omicron. The symptoms of people who got the third dose after, or even if they get Omicron, the disease is always very mild or asymptomatic because the vaccine can protect us and memory B cell can adapt to new variants. There is a group of people who have a problem, and these are fragile patients. They are really protected after vaccination. Maybe some will not be protected after vaccination. We can show you now an example with the vaccination of CVD, common variable immunodeficiency patients. You can see here that if you measure only antibodies in the serum, you find that 64% of the CVD patients make antibodies. But then we also measured the frequency of specific memory B cells, and we found that high affinity memory B cells were produced only in 24%, not 64% of the patients, meaning that when the antibodies will be gone, then these people will not have memory cells to be protected. So we really have to think about this because vaccines work, but they may not work in people with primary and secondary immunodeficiency. So thanks to the new tools and the high resolution instrument that we now have, we can really see not only the top of the iceberg, but also what is under. And we can measure the response to vaccines in a totally different way than before. Maybe this can be useful to accelerate vaccine development and also to identify patients who need special protocols and special treatment to protect them. One word of caution. This is just a recent joint statement from the European CDC and the EMA. And the problem is that we still have a high COVID incidence, rates of severe outcomes, and that's really low. This is because of the vaccines. But the problem is that the public was expecting no disease anymore, no contagion anymore. And also people think that the disease it's not so terrible indeed, because most of them will have a light and mild disease because they're vaccinated. This is bringing to what we call vaccination fatigue. People don't want to get, for example, the fourth dose. We have many patients with primary and secondary immunodeficiency who refuse the fourth dose, and this is a real problem because they are not protected enough. The last slide is to remind you that this actually is a slide from Rino Rappuoli to remind you that for many years we didn't live very long, but over the last 2 centuries our lifespan increased because of sanitation of the water, because of antibiotics, but also because of vaccines. Now COVID-19 has shortened our lifespan a few months because it's killed many older people. Now with vaccine we'll recover and we'll get through this. And I want here just to thank you all the people who did the work, especially Eva Piano Mortari, Sara Terreri, Anne Rodriguez, Cristian Albano, and Maria Giulia Conti, and my long-time collaborator and precious collaborator Franco Locatelli, Salvatore Zaffina, Isabella Quinti, and all the hospital, because I got blood from all my colleagues. Thank you.
Thank you very much, Rita. What a fantastic lecture, and really nice to see the team of ladies in the lab and female scientists. Thank you for championing that as well. So fabulous. So Rita, I'm gonna combine a few questions because there's quite a few coming in. And the first one is really about when you broaden your B cell repertoire over time, as you indicated, is this because of COVID antigen persistence, or does antigen persistence play a role? And how do we tie up, just a separate question, how do we tie up severe illness in older people if there's no waning of memory B cells? So first one is, how do we broaden the repertoire? Do we need antigen continuously? And then older people and waning memory. Thanks.
Yeah, thank you. Thank you for everything. And so the first question is, it has been demonstrated actually that antigen persists in the germinal centers of vaccinated people. So there are, not very active germinal centers. Germinal centers have a phase in which they really grow a lot, and then they are spent in a way, they are very small, but then antigen persists there, and memory cells will continue to circulate there and will improve their specificity. This has been demonstrated both in the disease and after vaccination, if you have seen. And there is just another recent paper, I think it came just yesterday again from Michel Nussensweig group, demonstrating that even the third dose increase again and amplifies the repertoire. We also started to measure the Omicron response, the response to Omicron, the memory B cell specific to Omicron that you can really see after the third dose. You could see few of them after the second dose. So probably there is a continuous modification of the antigen, of the response to the antigen, because the antigen is still there in our body. We really don't know. This is a very interesting question, and I really don't know the answer, is what happens with the other vaccines. With the other vaccines, we never had these tools, to measure antigen specific B cells with such a easiness. This is really easy, and we could do it in a lot of people from whom we know everything, for example, like the healthcare workers. So we don't know what happens in other response to other vaccine. If I'm vaccinated against tetanus, will tetanus persist? I really don't have the answer. But for COVID, yes, it works like this. As for the older people, I mean, the point is that we have started to study this, and if you see, after we become like 80 or so, there is a radical change in the memory B cell repertoire. So what happens is that most of the low affinity cells will be gone when you become very old, and then you are left only with the high affinity cells. But this is a real problem, because if you are left only with the high affinity cells, it means that you remember essentially what you have met many, many, many times. And then you are very good in answering to something but you're very bad in adapting to new things, and that could be one of the problems. And then, of course, older people have a lot of concomitant diseases also, so it should be studied better.
Okay, thanks, Rita. It's quite a lot of questions and little time. I'll try to squeeze at least two more.
Yeah.
So one question is, if T cell epitopes are added along with the spike, will it improve long-term memory B cells? And she meant CD4, CD8 T cells from N, M, and A.
If this would change the B cell response too.
If it would improve the long-term memory B cell response.
I think that what will happen is that you would have a even broader memory against other antigens. Antonio before showed that your T cells react against different antigens, and in reality, with the vaccine, we only use spike. So of course you will amplify the response, but the long-term memory, I think we have already like this, and in reality, B cells should do a primary work, which is to produce neutralizing antibody and avoid infections. Then T cells should kill the infected cells. So the antigen that they should see may be slightly different.
Yeah, and do you think there's a role for antibodies to the anti-nuclear protein? Do you think that's contributing to protection in any way?
I don't know. Honestly, I don't know, because we think that the most important thing for B cells is to act early, as a prevention.
Okay, and then just the last one, obviously antibodies are easy to measure, and you showed us beautifully that we are only measuring the tip. So is there a way to, a quick and efficient way to measure what's under the water?
Yeah, I have shown you, with antigen, it's easy. I mean, and now for us, it's a routine to measure antigen-specific cells. I think that would be, what I think this is one of the things that the pandemics has given to us, tools. We have learned a lot and we have new tools, and maybe this will help us a lot to develop new vaccines for other diseases and new pandemics.
Okay, thank you, Rita. I'm going to have to leave it there because of time.
Yeah.
But there's lots of questions, Rita, in the chat. If you're able to answer, please do.
Yeah.
Okay, thank you so much. So
thank you to you.
Thank you. So we're going to move on to our next talk, which is by Andreas Diefenbach. I'm just going to introduce Andreas. He's professor and chair of microbiology at Charité in Berlin. He's interested in understanding tissue-resident immune system and how it coordinates the adaptation of multicellular organisms to their environments. He's an elected member of the Berlin-Brandenburg Academy of Sciences and is recognized as a highly cited researcher in the field of immunology. So today he's going to talk to us about natural killer cell responses across the COVID-19 disease spectrum. Andreas, thank you for accepting our invitation and welcome. Thank you.
Good morning, everybody. Good afternoon, good evening, wherever you may be on this planet at this time of the day. Let me just start by thanking the IUIS and EFIS for giving me the chance to present our data here. And what I want to do in the next 20 minutes or so is discuss the role of natural killer cells during SARS coronavirus 2 infections. Now, before I go into the data, I would really like to acknowledge the people who did most of the work. And that is Caroline Tizian, a PhD student in my lab, Mario Witkowski, a postdoctoral fellow and clinician scientist, and Farzin Mashregi, lab, who we collaborated with very intensely, as you will see during the talk, who provided analysis of the single-cell RNA-seq dataset. And there were many other collaborators, in particular the virology laboratories at the Charité, Christian Drosten, Terry Jones, Daniela Niemeyer, and Leif Alexander, who is a professor and investigator at the Department of Pulmonary Medicine. Now, when we started to think about doing some work on SARS coronavirus infection, I think we were kind of driven by the view that this is, in most cases, a self-limiting mild infection that is being contained within the first 2 weeks following infection with the virus. And that also drove the view that most likely these early events then are decisive for determining the course of infection. And while, of course, much work had been done on T cells, on B cells, in terms of protecting, providing long-lasting protection, we felt there was a gap in our understanding of other players in the immune response, such as innate lymphocytes, IL-6s, natural killer cells. And I want to make one additional point. When it comes to innate immunity, of course, you will see often cartoons like this, that an appropriate early type 1 interferon response is central for being essential for deciding whether there is a beneficial course of the disease, because the early type 1 interferon response would then instate a antiviral state of infected cells that would decrease viral load and will lead to a mild disease, whereas a somehow delayed type 1 interferon response will lead early on to a very high load of virus and then viral persistence, inflammation, and eventually severe disease. Now, this is, of course, a bit of a oversimplifying scheme, as there is clearly also regulators of interferon responses, of early immune responses, from which we don't know so much yet. And I think that is, you will see, relevant for also how NK cells actually respond to virus infections. Now, let me quickly sum up some of the very basic facts about natural killer cells. Maybe not all of you think about this all the time. These are clearly bone marrow-derived lymphocytes. They are cytotoxic cells, hence the name, that have a propensity to recognize tumor cells and infected cells, in particular virus-infected cells. They are part of the innate, directly acting immune system, so they can actually recognize target cells and kill them within minutes or hours. And there has always been a discussion that NK cells may have been developing during evolution by being an important barrier to virus infections. And the people who have NK cell deficiencies are predisposed to a more severe course of viral infections. And that is also supported by an array of mouse models that show that at least the early phase of a virus infection is initially dominated by NK cell mediated control, and then later on adaptive components of the immune system take that over. Another link to virus infection is that the type 1 interferon module is one of the probably most potent activators of NK cells, often in an indirect way by inducing other types of cytokines. But in any ways, that is a further link between NK cell activation and virus infection. But there's also some maybe not so well recognized functions of NK cells that actually attracted our attention in the context of COVID, because NK cells have been discussed to play a important role in also regulating immune responses itself. So, for example, work from Maria Pia degli Esposti's lab some years ago actually showed that they can control overzealous myeloid cell responses by probably directly killing or dampening the function of myeloid cells, thereby actually preventing things like a cytokine storm or over activation of the myeloid system that comes with collateral damage. And another interesting study actually linked NK cells to antifibrotic functions. That was work performed in Scott Lowe's lab, showing that NK cells can actually directly recognize cells that become fibrogenic and thereby positively influencing fibrosis development. And obviously, both of these qualities could be involved in COVID-19. So we thought that would be a good idea to give this a look. And I've summarized this here again. I mean, in particular, the increased innate inflammatory output and the hypercytokinemia could also be reflecting some failure of the immune system to provide basically an immune activation checkpoint for the myeloid system. Now, what we started with is a bit of an epidemiological piece of data that was supported by Terry Jones, where we basically looked at a very big collective of patients that came to the clinic and where we had an immediate NK cell count. And we stratified these patients in two groups, one that had an NK cell count of more than 40 microliters and one that were below this threshold. And when we then basically consecutively measured viral load, what you can see is patients with a higher NK cell count have a reduction in viral load over time, and those with a lower NK cell count to a much lesser extent. And obviously, this is clearly not many patients who present with such lower counts, but one has to say, when we do same analysis for other types of blood cell populations, such as B cells or T cells, we don't see such tendencies. Now, this basically gave us reason to actually ask, do NK cells really recognize virus-infected cells? So what we did is we used two cell lines that you can infect with SARS coronavirus, two Vero cells and CADO3 cells, a lung epithelial cell line. And what you can see in particular, fully activated NK cells, those dependently basically curtail virus replication. So the more NK cells are in the system, the less the virus can replicate. And as NK cell biologists, we were wondering, is the activating receptor systems being involved in this? And what we did is blocking experiments in such assays. So to basically see whether blockade of a or multiple activating receptors would actually lead to enhanced virus replication. And single blockade of activating receptors did not really have any appreciable effect. But when we basically combined receptor blockade of 3 receptors here, for example, we did see a significant effect on virus replication, indicating that probably redundant signaling via a range of activating NK cell receptors contributes to recognition of virus-infected cells. And NK cells are also express inhibitory receptors, many of which recognize class 1 MHC molecules. So we tested whether further inhibition of class 1 MHC molecules would lead to even better virus control, and that's shown here. But you see that had no effect, so that we don't think that NK cells are appreciably inhibited by class MHC, at least in these in vitro assays of NK cell mediated control of virus replication. Now, these experiments were, of course, all performed with NK cells from healthy donors. So we tested in comparison NK cells from patients with severe COVID. And what you can see is that they are quite impaired in their propensity to actually reduce virus replication in infected target cells, both in the varicella and in the CALO cell system. So we wondered what's going on with NK cells during COVID. And for this, we actually recruited a larger cohort of patients, healthy donors, patients that have actually a flu-like illness, a mild disease, but not caused by SARS-CoV-2. We had a rather substantial group of mild disease patients, so that were seen in an outpatient setting, and then hospitalized patients with moderate or severe COVID-19. These patients were repetitively sampled with up to 11 samples per patient, and this ranged from basically their time point of symptom onset when they were first seen by a doctor up to 36 days after symptom onset. Now, when we looked at the expression of the cytotoxic machinery, what we saw is actually that NK cells made an activated impression, basically. So they had increased levels of both perforin and granzyme compared to healthy donors, and that was very similar to other types of respiratory non-flu-like illnesses. So we expected that NK cells may be activated and may be in the position to kill target cells. But when we performed killing assays, what we saw is that NK cells, in particular from hospitalized patients, but also as a trend, patients that presented in a outpatient setting had impaired killing of target cells. And we didn't see this with patients with other flu-like non-SARS-CoV-2 infections. So despite the high expression of molecules that are linked to cytotoxicity, we saw a very much diminished killing activity, and we wondered why would that be. Killing requires degranulation of cytotoxic granules after contact with target cells, and indeed a degranulation measured here as CD107 surface expression after co-culture with target cells was much diminished and really a function, if you wish, of the severity of disease. And that was also, visible here, so that this was really only seen for hospitalized patients with a moderate or severe course. We did not see this in mild patients, and we did not see this in patients with other flu-like illnesses. Now, one of the key points of NK cell cytotoxicity is that they make conjugates with target cells, and this can be measured in these kind of co-culture assays. And as you can see, NK cells obtained from severe COVID-19 patients had a strong diminishment in the formation of such conjugates between NK cells, and this may be a reason why NK cells cannot recognize their target cells as well. Now, to put this basically a bit on a molecular scale, we obtained a single-cell RNA-seq atlas of about 90,000 NK cells only. And I don't have the time to go through all the details of the clustering here, but maybe suffice it to say that we recovered the recognized large clusters of NK cells that we also find in the disease setting. There were some significant shifts. So, for example, this population up here, which represents proliferating NK cells, shown here, is significantly increased during severe disease, as is this cluster 3 here, which reflects terminally differentiated, maybe anergic type of natural killer cells. Now, when we did pathway analysis on our data set and also which functional pathways are enriched in NK cells during disease, what we found is that clearly NK cells showed signs of cellular activation by the interferon system, as already predicted by their high expression of granzymes and perforin, for example, as you can see here. So clearly, although they are functionally impaired, they do express high levels of a type 1 or type 2 interferon response. Now, then we got a bit of a puzzle, or puzzling finding, that they're also enriched for a pathway that's related to IgA production. And initially, I was ready to discard this finding, but then we thought a bit about it. And as you know, isotype switching to IgA depends on the cytokine TGF-beta. And many of actually the molecules are target molecules, in fact, of TGF-beta in this cluster. So we wondered, is it then so maybe that TGF-beta is being produced and may have inhibitory functions on NK cells? For that, we wanted to look whether TGF-beta response signature is enriched in NK cells, and there is no such data in the available databases to us, at least. So we obtained a single cell atlas of normal NK cells that we stimulated with TGF-beta, and then we took that signature and superimposed it on our patient's data set. And what you can see is indeed that there is also a TGF-beta signature in healthy normal patients, which is consistent with the fact that there's always a bit of TGF-beta in the serum, as you will also see later. But these signatures are clearly enriched in patients with severe disease, as are the genes that are negatively regulated by TGF-beta. And that's something we find already quite early during disease with a really high significance. Now, when we mine the data further, one of the pathways, or the group of pathways actually that was modified in severe disease were those that were related to cell-cell adhesion. So one of the major modules for NK cells to fulfill that cytotoxic activity, and even some single integrins we could identify, such as integrin beta-2, that were consistently lower expressed in patients with a severe disease. Now, one could of course wonder, is this maybe all not so exciting because it might happen in any type of infection? So we actually reanalyzed several other data sets. I show you here one from a comparison of COVID patients and influenza patients. We isolated NK cells from that data set, and then we overlaid our TGF-beta signature. And what you can see here is that there's a clear enrichment also in this data set for a TGF-beta signature, but that was not seen in patients' NK cells that were infected with influenza virus. So there seems to be some preference to TGF-beta signatures in NK cells in the COVID setting, which was not found in other infections. And we confirmed this on a lung cell atlas that I will discuss with you in a second, where we saw basically the same effect, that we only saw this in COVID patients to have such a dominant TGF-beta signature. Now, where does the TGF-beta come from? I don't think I'll have a final answer for you, but we did indeed reanalyze a quite extensive lung cell atlas that was performed by colleagues in the neuropathology and pathology departments at the Charité. And what you can see on first sight is that there is clearly some clusters that upregulate genes that are either associated with TGF-beta function or TGF-beta genes itself. And maybe this is a bit more instructive. So in particular, fibroblasts, endothelial cells, but also myeloid cells had increased expression of TGF-beta 1 here, but this was very similar for other TGF-beta related genes. And one incidental finding was that the cells that actually have the highest levels of this expression are those that are infected with the virus, because we detected viral RNA in our single-cell RNA-seq data sets. Now, would that also be something we can measure? And we did that. So we basically performed ELISAs from serum of patients at week 1, 2, and 3. And what you can see is that indeed the patients that had reduced NK cell function, so the hospitalized patients, they had higher levels, significantly higher levels than those that had a mild disease or that had a non-SARS-CoV-2 flu-like illness. And that was something we saw up to 3 weeks. And then after 3 weeks, it seemed to be that the levels of TGF-beta level down again are not distinguishable anymore to the mild disease patients. And as we had found that NK cell function in terms of curtailing virus replication was diminished in severe COVID patients, we wondered, could that maybe then be a direct effect of TGF-beta on these cells? And that would have, that's not basically a totally novel finding. Already in '86, no other than Tony Fauci showed that actually transforming growth factor beta can inhibit NK cell function quite profoundly. And that is entirely true. Also for the recognition of SARS-CoV-2 infected cells, because when we pre-treated healthy NK cells with TGF-beta and put them then in the virus replication assay, you can see there is no inhibition of virus replication whatsoever. And also killing of target cells is dramatically diminished in those cells. And we confirmed this for a range of NK cell functions. So TGF-beta is really a major inhibitor of NK cell functionality. Now, that we could then also replicate by taking serum from patients who have COVID and put that on healthy NK cells. So that the question was basically, is the activity in the serum enough to inhibit NK cells from healthy patients? And you can see here, indeed, serum from severe COVID patients reduces degranulation, and it reduces also the expression of transcription factors that underpin the activating function of NK cells. And finally, we performed the same assays, but blocked in the serum when adding it to the target cells and to the NK cells. We blocked basically TGF-beta with antibodies. And you can see that did actually at least partially restore virus control and restored NK cell function in terms of degranulation and in the expression of transcription factors. So I think that's what I wanted to share with you. I want to summarize here that NK cells can control SARS-CoV-2 replication in infected target cells, at least in vitro. They do this by likely redundant signaling through a variety of activating NK cell receptors. But SARS-CoV-2 seems to promote an untimely early TGF-beta response, which seems to be a hallmark of COVID-19, because we didn't find that in non-COVID respiratory diseases. And this kind of propensity or this kind of close link of SARS-CoV-2 to trigger TGF-beta response is actually now supported by a variety of studies, and I'll show some here. One is from Andreas Pickelmeyer's lab at the Technical University in Munich, who showed that the open reading frames 3 and 8 of SARS-CoV-2 actually have a very close relationship to either TGF-beta signaling or to the latency complex, and releases more TGF-beta and thereby promoting TGF-beta signatures. Another study, which is not published yet, from Eva Harris's lab, shows that basically vascular leakage that is being triggered by the spike protein of SARS-CoV-2, as shown here. So this is an intratracheal administration of spike protein, and then looking at vascular leakage in the lungs, and you can see the spike protein does actually trigger quite some disturbance of vessel integrity. And that was entirely dependent on TGF-beta signaling. And the model that the authors propose is that SARS-CoV-2 spike protein actually triggers release of TGF-beta and enhances TGF-beta signaling, which then in the end leads to endo and epithelial hyperpermeability and vascular leak. And a last study that I want to mention is one that shows that the nuclear protein of SARS-CoV-2 actually stabilizes and enhances SMAD3 activation, which in the end then enhances TGF-beta-induced transcriptional protein. So then this early and maybe at this early stage inappropriate expression of TGF-beta leads to a long-term reprogramming of NK cells and limiting potentially NK cell-mediated virus control. And this may indicate that interference with this type of early TGF-beta activation and signaling could be a therapeutic target. And there is one company that has trying to explore this, Oncotelic is the name of the company, who has an antisense drug against TGF-beta. And they have used that in one small trial, which they published on the website, showing that patients that actually get standard of care treatment plus the TGF-beta inhibitor show actually reduced mortality and increased viral clearance. And now this is what I want to stop with. Again, I want to acknowledge people, Mario, who really initiated the study and pushed it forward together with Caroline. And we would have not been able to do this without the really exciting collaboration with Fars and Regis Lab. These are our funders, and I want to especially mention this one here, which is an initiative of the German federal government to give money actually to all universities to work together to fight COVID, and particularly to explore immune responses, which actually did really did trigger quite some collaborative spirit across our country to actually understand what are the immunological features of this new kind of virus infection. Thank you very much, and I'm happy to take any questions you may have.
Thank you very much, Andreas. That was a terrific talk with a fascinating finding, especially the one on the role of TGF-beta. In fact, we have actually several questions that are going in the direction of understanding how TGF-beta is triggered and what is the role of TGF-beta. I will just pick one because we are quite short in time, but I really ask you if you can answer on the Q&A. But one question is whether thrombotic events during severe COVID has any relation with TGF-beta, and what can be the trigger of TGF-beta release in the context of SARS-CoV-2 infection?
I didn't get the initial one. So what can trigger
what can trigger the TGF-beta secretion, and more importantly, whether there is any correlation between TGF-beta and thrombotic events in SARS-CoV-2?
Yeah, I mean, we haven't looked at this at all. I'm also not aware of a lot of data. So I think the best data that goes to endothelial cell function is this still unpublished work of Eva Harris's lab that shows that spike protein itself basically can disturb endothelial and epithelial resistance and thereby triggering basically vascular leakage. Whether somebody has explicitly looked at TGF-beta and thrombosis formation, I'm not aware of. Yeah.
Yeah, but as I said, there are many questions, not only on the TGF-beta, for instance, whether the long-term changes in NK cells affect infection with other viruses. That is something I think you will follow up.
Yeah, exactly. I mean, I don't know that, but that is a interesting question. We really did see a long-term reprogramming even after times when virus was cleared. In cases were still somehow down in their function. So that is something we would really now be curious about. Is that something that affects virus responses, or is there some type of hidden memory somewhere that may even increase? So that is something we would just really like to know.
The memory, in fact, there is also a question of memory. So please have a look at the Q&A.
Yes,
there are interesting questions from our attendees. Thank you again, Andreas, for coming and joining and showing us these interesting data. And now it's really my pleasure to introduce the next and the last speaker for this part of the webinar, Professor Akiko Iwasaki, who is a professor of immunobiology at Yale University and investigator of our youth's medical institute. As you know, she has really gave a fantastic contribution in the field of immune defense mechanism against viruses on mucosal surfaces. And more recently, she's really leading a Yale COVID recovery study, which aims to determine the changes in immune response of people with long COVID. And this is actually the topic of her talk. So thank you very much, Akiko, for joining us today, and we look forward to your talk.
Thank you, Federica. Hello, my name is Akiko Iwasaki. Today, I'd like to talk about immunology of long COVID. Over the pandemic, my laboratory has focused on trying to understand the immunology of acute COVID, as well as more recently long COVID. We've seen that the variety of outcome of COVID-19 can happen from asymptomatic infection all the way to fatal disease, and some people suffering acute disease while others have prolonged disease, the long haulers. We've shown that, for instance, by comparing the immune response of moderate, severe, and fatal diseases in hospitalized patients, that there is a variety of immune responses that are distinct among these groups, such as the viral load being maintained at high levels in severe COVID cases and delayed onset of neutralizing antibodies in the case of lethal COVID. We've also seen direct infection of the neurons within the CNS of COVID patient autopsy, as well as in mouse models, indicating that neurotropic infection can happen after a respiratory infection with SARS-CoV-2. We've also focused on the difference between male and female patients' immune responses and demonstrated that early immune responses that occur in female patients, such as the activation of T cells, are not happening well in male patients. And especially with age, male patients lose their ability to stimulate T cells, and this correlated with worsening of the symptoms. We've also been looking at cases of COVID-19 in pregnant women, in one case leading to a fetal loss due to direct infection of the placenta, syncytial trophoblast with SARS-CoV-2. And in other cases, even without direct infection of the placenta, we've seen that there is significant transcriptome changes that occur in the placenta. And so this illustrates the diversity of immune responses that can occur after a single infection with the virus. Today, I'd like to focus my talk on the long COVID. So what is long COVID? Well, long COVID it can happen in a fraction of people who have either severe disease, who are hospitalized, as well as non-hospitalized patients. And I'll get into this more in detail. Before I get into long COVID, however, I'd like to emphasize that there are multiple different outcomes from other infections that resemble long COVID, such as the post-acute infection that happened with Ebola virus, dengue virus, polio, the original SARS virus, and a number of other viruses listed here, which accompany a fraction of patients who have prolonged symptoms over months to sometimes years. And it's not completely restricted to viral pathogens either. If you look at other non-viral pathogens such as Coxiella, Borrelia, and Giardia, there are also reports of prolonged symptoms that happen after these types of infections, leading us to believe that long COVID is a part of the large collection of post-acute infection syndromes that occur. So if you look at the different types of infection, this is just indicating the Epstein-Barr virus infection cases. You see that time since mononucleosis is indicated on the x-axis, and the y-axis indicates unrecovered participants. So within about 2 months of mononucleosis, you have close to even 40% of the patients having still unrecovered from the infection. This percentage declines over time, but even at 24 months, there are 4% of the patients still unrecovered from EBV infection. So this is indicative that infections like EBV can lead to prolonged symptoms, and in, unfortunately, a handful of patients, this extends to over 2 years. And also with SARS-CoV-2 infection, if you look at, again, this time since COVID-19, this time the x-axis are in days. You can see that some patients failed to recover from the infection. So at least having 1 of 12 common symptoms with long COVID, you can see that a fraction of patients still suffer from that 125 days out. Similarly, self-reported long COVID that results in activity limitation. So these are severe forms of long COVID, still maintained fairly at high levels, hovering over 5% at 168 days, indicating that these types of prolonged symptoms also occur after SARS-CoV-2 infection. So the long COVID definitions differ slightly between different organizations. So I've listed 3 commonly used definitions of long COVID from the WHO, the CDC, and NICE. And you can see that the definitions differ slightly between these. In the WHO case, prolonged symptoms lasting at least 2 months within the first 3 months from the onset of the original symptoms, whereas the CDC definition is 4 weeks or more. NICE definition is more than 12 weeks. So, currently there is no universal global definition for long COVID, and this makes it difficult to measure the prevalence of long COVID across different countries. As I mentioned earlier, there are different paths to getting long COVID. In some cases, severe critical COVID, acute COVID, that led someone to be hospitalized. These people, the survivors who are discharged, do tend to have prolonged symptoms, and between 50 to 75% of these people suffering months of long-term symptoms. And many of these are likely related to some organ damage that resulted from the acute phase of infection, or potentially even with the treatment regimen that they had received. And then there are these milder cases which did not require hospitalization. These patients with or without symptoms could also lead to post-COVID-19 fatigue syndrome, which are sort of new onset unexplained symptoms, including fatigue, post-exertional malaise, neurocognitive deficit, and some respiratory symptoms. And currently there are over 200 different symptoms being reported for long COVID. And again, there are no universal definition of long COVID. However, there are many symptoms that affect organs, and essentially we haven't found a single organ that's being spared by the different symptoms that have been reported thus far. So how does long COVID happen? Well, this is a major question that my laboratory, in collaboration with a large team of investigators around the world, are currently investigating. And potentially the first hypothesis is that there may be viral reservoir or PAMPs or protein antigen from the virus that is prolonged remaining somewhere in the tissue. That could activate chronic stimulation of lymphocytes that are reactive to the antigens, or pattern recognition receptors that are stimulated by RNA, or potentially some forms of self-nucleic acids. Second possibility is autoimmunity. There may be autoreactive T and B cells that are being stimulated as a result of the acute phase of the COVID-19 disease. And of course, once these autoreactive cells are generated, it's very difficult to get rid of them. Third possibility is dysbiosis or reactivation of latent viruses such as Epstein-Barr virus, which I'll get into. Also tissue damage that was caused from the infection or host response to infection, when not repaired properly, could result in prolonged symptoms. So for instance, there are several studies now supporting the viral, at least RNA and or protein presence in various tissues, including the intestine, the lymph node, and fat tissue. There's also evidence for EBV reactivation during the acute phase of the infection as one of the 4 predictive factors for prolonged long COVID symptoms. There have been studies published showing that autoreactive antibodies against G protein coupled receptors can be found on all of the participants that they've examined who had COVID before. There's also the same paper in Cells by Su et al. Showing that autoreactive antibodies to lupus antigens are correlated with or one of the predictive factors for developing long COVID. And finally, I'm going to talk about a study under review that demonstrates that distal infection, meaning the respiratory infection, can have neurological impact on the animal, even with an extremely mild infection. And this preprint demonstrated that dogs can detect long COVID patients based on sniffing of T-shirts from such patients. And the identification rate, it was 51%. And then in the control, there was 0%, meaning that it's a very accurate and relatively sensitive measure, meaning that the dogs can really distinguish long COVID patients from non-long COVID people, indicating that there may be volatile molecules that's being emitted by long haulers that can be used to distinguish disease state. So people have measured a variety of immune factors in circulation of people with long COVID, in this case in red, versus those that are matched controls who've had COVID but don't have long COVID in blue. And this paper in Nature Immunology demonstrated that there seems to be prolonged interferon beta and interferon lambda 1 in patients who have long COVID at the 8 months time point. And in fact, interferon beta was the top feature that was identified to be uniquely supporting the long COVID status in this particular paper. Autoantibody to variety of G protein coupled receptors are found in long COVID patients. As I mentioned, this study by Walukat et al. Demonstrated that all 31 participants who have had COVID prior had between 2 to 7 different anti-G protein coupled receptor autoantibodies that were functional in vitro, stating that perhaps these kinds of autoantibodies can dysregulate the homeostatic function of these GPCRs, which control heart rate and other essential parameters for host physiology in people. And as I alluded to before, the Su et al. Cell paper identified 4 PASC, anticipating risk factor. PASC is another word for long COVID. And in this study, they monitored longitudinally people who've contracted COVID from initial diagnosis to 2 to 3 months, and found that there are 4 anticipating risk factors, 2 of which I've already mentioned, Epstein-Barr virus, viremia, and autoantibodies, but they also identified type 2 diabetes and SARS-CoV-2 RNAemia as 2 other risk factors during the diagnosis that predict long COVID. In this study, they also found many other interesting features, such as dysregulated circadian rhythm that's found in patients who are presenting neurological symptoms, and then cortisol levels being lower in those presenting respiratory viral symptoms. So this indicates that these types of physiological circuits are maybe being affected in these long COVID patients. So in the remaining time, I'd like to introduce you to our preprint, which is on bioRxiv right now, which we had the fortune of working with Professor Michelle Monge at the Stanford University and her team to understand the impact of mild respiratory SARS-CoV-2 infection on the CNS. And this study really examines the potential impact of respiratory-only infection on CNS. And we designed this study in such a way that animals are transduced with the viral entry receptor humanase 2 intratracheally, and this enables the mice to become only infected within the lung, nowhere else. For instance, the brain is completely devoid of SARS-CoV-2 because there's no entry receptor. And these animals, when given a small amount of SARS-CoV-2, they have absolutely no weight loss, no sickness behavior, nothing we can even distinguish as being infected. But we know they are infected because we can find SARS-CoV-2 nucleocapsid stained antigens in the lung after 7 days. So using this strategy, we wanted to understand what if there if there's any impact of respiratory-only mild infection in the CNS. So these are the cytokines that we measured from the serum on top or the cerebrospinal fluid on the bottom. And the redder the signal, the more cytokine we find. So 7 days or 7 weeks post-infection. And you can see that even at 7 weeks post-infection, there's significantly elevated levels of cytokines and chemokines that are found in the serum and in the cerebrospinal fluid, including CCL11, which has been implicated prior to be neurotoxic. So we've examined the brain of these animals carefully, and what the team found is very interesting in that in 2 different strains of mice, so the CD1 or the BALB/c mice, we found a significant elevation of reactive microglia in mice that have been infected with SARS-CoV-2 at 7 days post-infection and also at 7 weeks post-infection. So this data suggested that there is a significant impact on the CNS of a respiratory-only viral infection. And next, we wanted to understand whether this has any relevance in humans. And so in collaboration with Dr. Abindranath at the NIH, we've looked at patients' autopsies from people who've had mild COVID and died of other causes. And in this case, again, we see particularly the white matter having significant reactive microglia in the patients, but not in the non-COVID control, or patients having much higher levels of reactive microglia than the control people who died without any COVID. We've also seen other impairments within the CNS. So this is looking at the hippocampal neurogenesis and at 7 days post-infection or 7 weeks post-infection on the bottom. You can see that compared to control animals, the neurogenesis in the hippocampus is significantly impaired. And we've also seen correlation, as I mentioned, the CCL11, which has been shown to have an impact on neurocognitive functions, are elevated in the mild COVID animal CSF, even at 7 weeks post-infection compared to control animals. And we also find elevated levels of human serum CCL11 in long COVID patients with brain fog compared to the without. And as a result of reactive microglia, we also see loss of oligodendrocyte from the white matter. And you can see here, there's a significant reduction even at 7 weeks post-infection. And because of the loss of oligodendrocyte, there is reduced myelination of the axon. You can see here clearly, even at 7 weeks post-infection. So these results indicated that even mild respiratory-only SARS-CoV-2 infection leads to prolonged changes in the CNS at 7 days and 7 weeks post-infection, and that autopsies results show similarly increased reactive microglia in the white matter of patients, and that the direct infection of CNS is not required to induce significant CNS pathology. So I'd like to end by thanking the members of my lab who all contributed to a variety of projects that I was able to mention today, as well as the Impact Yale team who assembled patient cohort studies, and the Mount Sinai Long COVID team, without whom we can do any of the human research that we're doing currently. And then also the Yale team of long COVID patients that we're also collaborating together, along with immune profiling data analysis team, as well as the Stanford collaborators that I mentioned today. And I also like to thank SurvivorCore and Patient-Led who helped design many of these studies, as well as the funding agencies. Thank you for your attention.
Thanks so much, Akiko. Always really, really great to hear your work, and so much data and so much for us to discuss. Time is short, unfortunately, so I'm gonna lump 2 or 3 questions together because we only really have a couple of minutes now. But the first one is, what do we understand about, or what do we know about the effect on the fetus when the mother contracts COVID in the first trimester? And second question is could autoimmunity be due to homology between the T cell epitopes from SARS-CoV-2 and human self-peptides? And therefore, is vaccination a risk in terms of inducing autoimmunity? Let's start there.
Wow, thank you very much, Faith. So to begin, the first question about the risk of pregnant women getting COVID, there are now multiple studies that looked at this and in humans, and they found that the risk for developing more severe form of COVID is higher in pregnant women compared to non-pregnant women. And that's also consistent with other pathogens like influenza. So pregnancy does bring some risk factors for infectious diseases, including COVID. There's also been reports of preterm birth as well as stillbirth in women who contract COVID in the first and second trimesters. So there's also impact on obviously the fetal development and fetal health in the women who are pregnant, who are infected with COVID. And that is why we've been saying that women should get vaccinated, as much as possible and to avoid these kinds of consequences. The second question about homology of T cell epitope from the SARS-CoV-2 spike to host protein. Yeah, that's something that we've been very interested in looking at. We have done a lot of autoantibody work where we've been able to identify autoantibodies that some of which have some cross-reactivity to the anti-spike to host proteins. We haven't extended to T cell epitopes yet, but the fact that, so many hundreds of millions of people are vaccinated and boosted, and we haven't seen autoimmunity in the vaccinated people, I assume that there's very little chance of that happening. During the infection, however, there may be inflammation that accompanies activation of autoreactive cells.
Okay, thank you, Akiko. This is a longish question. Lots of positive comments, by the way. Really, your talk was well appreciated. But Rahul Gupta asks whether changes in microbiota and epigenetic changes in the endothelium that lines all organs could be another hypothesis behind long COVID. And because dogs can sense both COVID and long COVID patients and volatile organic compounds only due to the degradation of sweat by microbiota, do you think that microbiota changes can also contribute to long COVID?
Yeah, absolutely. So microbiome change is one of the 4 hypotheses that I discussed, but I didn't really elaborate on the microbiome changes. There are now studies showing that microbiome of long COVID patients are indeed distinct from healthy people, and that there are certain bacteria that are enriched that are commonly found in the long COVID patients. Whether their metabolites are leading to the dog's sensing of these people, or what actually is being sensed by these dogs, it's unclear right now. But I like the idea of the microbiome contribution to the volatile organic compound release from the sweat gland. That was my first hypothesis, but the authors seem to think that it's mostly the SARS-CoV-2 antigens because the dogs were trained on the supernatant of SARS-CoV-2 infected cells.
Wow. Okay. So I'll just ask the last one, and then maybe if you have time to answer a few more in the chat, that'd be really appreciated. So the last one's from Yanette Valdez, who asks, what are the most important interventions that you think we need to begin to address long COVID?
Great question. So we are looking at the impact of vaccinations and other measures like antivirals and monoclonal antibodies. Unfortunately, I haven't come across, the vaccination part is even controversial, even though there are like about a dozen papers showing some impact of vaccination on prevention of long COVID. The most recent papers show very little impact of vaccination. So we are still kind of struggling with the understanding of what can we do to intervene or prevent. I would think the vaccination is absolutely key because if you can prevent the infection, you can prevent all consequences. Mask wearing, avoiding indoor crowding areas. I mean, these are all the measures that we've been doing over the last years, but I think we need to keep doing it.
Yeah, great. Thank you. Thank you so much, Akiko. We really, really appreciate it. Really informative and fantastic data. I'll just refer you to the questions if you can. And thanks again for joining on behalf of all of us from all over the world who are listening in. So now I hand over back to my co-chair, Federica, to take us to the next session. Thank you.
Thank you very much, Akiko, and thank you, Faith. We are a bit late, so I won't take a lot of time. We are just taking a few minutes break before the last very interesting session on a panel discussion that will be moderated by Apoorva Mandavilli from the New York Times, and has a lineup of panelists who are really going to touch upon the problem of current state challenges and prospect of COVID-19 vaccine. Just 2-3 minutes of break while we are running a little video that was done with the contribution of national societies, regional organizations, young atheists, Immunopedia ambassadors that are going to be displayed on social media, YouTube channel, then also on Twitter, Twitter and Facebook, follow all these activities that have been running under the hashtag Thank You Immunology. 2-3 minutes break.
To me, immunology is an amazing field of study.
And the component of immune system can be engineered or utilized to kill infectious organism or tumor.
My favorite part of working in a lab is when I correctly diagnose diseases and therefore help clinicians treat patients as soon as possible.
Gut bacteria are the key to a healthy immune system. Like they say, you are what you eat.
My favorite fact about vaccination is that vaccination is the most important thing we can do to protect ourselves and our children against illness.
Some viral infections have been overcome or eradicated through production of vaccines, like smallpox in children, as well as rinderpest in cattle.
I encourage young people to study immunology because it's an exciting field in human medicine, and it provides answers to the prevention and cure of infectious diseases.
Your coronavirus vaccine questions answered.
How do vaccines work?
Hi, everybody. We're back for the second part. Please continue to watch the video online. But because of time, we're going to stop there. And it's really, really my pleasure to introduce the panel for the next session. Our chair of the panel, our moderator, is going to be Apoorva Mandavilli. Apoorva is a reporter for the New York Times. She focuses on science and global health. Currently covers the pandemic, vaccination, WHO, amongst many others. She's the founding editor-in-chief of Spectrum, and she joined the Times in May 2020. Also on the panel, we're gonna have Emma Hodcroft, who's formerly a biologist, who trained in the Texas Christian University. She's focuses now 100% on SARS-CoV-2 research and science communication that's related to the pandemic. Our third panelist is Rita Barbouche, a professor of immunology and assistant dean of research at the Medical School of University of Tunis, Mana. He's head of clinical immunology department and a director of research lab at the Institut Pasteur de Tunis in Tunisia. So thank you, Rita. And finally, I have Florian Krammer, He's Mount Sinai Professor of Vaccinology at the Icahn School of Medicine, and his work focuses on understanding the mechanisms of interactions between antibodies, viral surface proteins, and how this leads into vaccines and therapeutics. So a really, really impressive panel. And without further ado, I'm going to hand over to Apoorva to kick off our discussion. Thank you very much.
We just also welcome Skip Virgin, who is Executive Vice President of Research and Chief Scientific Officer at Biotechnology, and is leading research into SARS-CoV-2 monoclonal antibodies. So thank you all for joining this panel discussion, and Apoorva for leading it.
Thanks very much for having all of us, and I'm really excited to get the conversation going. So I thought I would start with a couple of questions directed at specific panelists, and then, if other people have things to say, they should join in. But my first question is for Emma Hodcroft. You and I have spoken a bit about virus evolution over the past couple of years, and I wanted to ask, what is the one thing that you think is so difficult about this particular virus, or challenging about this virus and its evolution, that makes designing vaccines difficult?
So hi, Apoorva, it's lovely to see you again. And thank you everyone for the invitation. It's really wonderful to be here. I think right now, the immediate challenge, both in pandemic response and in vaccine design, is the unpredictability of this stage of the virus. SARS-CoV-2 is a completely novel virus, and it's obviously spent a fair bit of the last 2 years adapting to humans, and that's included things like increasing its transmissibility, moving where we see a lot of the replication in the body to the upper respiratory system, as we see with Omicron, and of course getting around immunity, as we've also seen really significantly with Omicron. And what we don't know going forward is this kind of the peak point? Are these the main changes that SARS-CoV-2, the big jumps that it can make? Or are there still other big changes that it could make out there that could really influence both how we react to the pandemic, and also what's going to circulate in the future? Because I think one big unknown right now, for example, is, should we be pursuing adding something like Omicron to our vaccine? Should we be starting to use that as a first vaccine once it becomes available? Or will we see something completely new in the next 6 months that it would, there's no point getting into Omicron now, we're going to see something different. And I think that really poses challenges across the board, but in particular for figuring out a vaccine strategy right now.
Thank you for that. And Florian Krammer, I'm going to ask you a very difficult question, sorry. How long do you think the immunity from the current vaccines will last? And how does that compare to other viruses that you worked on?
Yeah, that's an interesting question, because if you talk to us, we're talking about immune responses and immunity, we talk about what we measure, right? And I think those immune responses that people get are actually long lasting, right? So it's not that the immune responses from after the first 2 vaccinations with mRNA vaccines, for example, disappear. We have also now seen that if you get infected, your immune response to that infection is long lasting, right? The problem that we see is that, of course, there is a big response after immunization or after exposure to the virus. And then there's some constriction of that immune response, and it goes down to some degree. And at the same time, as that happens, the virus started to change, right? What Emma just said. And with Omicron, SARS-CoV-2 figured out a way to circumvent most of our neutralizing antibody response. It's still good with binding antibodies and T cells, but that, of course, also didn't help. So I think there is still a large amount of immunity that is there, but it always depends on what are we protected from, right? The protection from infection with the current vaccines is not long lasting anymore, because antibody titers fall and the virus circumvents our neutralizing antibody response. The protection from severe disease is still very high. Just because you add now an anamnestic response, T cells are coming in, memory B cells are reactivated, and they have a lot of time to actually attenuate the disease while it's progressing, right? So I don't think immunity is disappearing, and I think what we see is pretty normal. But of course, we don't see what we saw in the beginning, which was solid protection, basically also from infection with the vaccines against the ancestral strain.
But to sort of really push you on that, how long will it last? If we don't see any crazy new variants, and if we stick somewhat close to the ones that we've seen already, Omicron, Delta, how long do you think potentially the protection from the current 3 doses that a lot of people have gotten, how long would that last?
I think that might last for years, but again, it has to do with, I think it would be better to have now a booster vaccine that's variant specific. Again, the problem is that we don't know what's coming next, right? Maybe Omicron is going to be replaced by a variant that's completely different. If it would be a linear evolution, it would be much easier to now say, let's update it and then we're good. But I think if the virus doesn't substantially change and we get an Omicron, and Omicron is a big family of viruses or strains by now, an Omicron specific vaccine that might last for a long time if the virus doesn't change again. I can't tell you in how many years, but I think this baseline immunity that we acquired that protects many of us against severe disease might be something that stays for a very, very long time.
Okay, thank you for that. And, oh, yes, go ahead.
So perhaps to start making things a little more interesting, I think there's what you can measure and how long it lasts. And then there's, does it matter? And then there's clinical trials, and then there's the real world. And I am not as confident that there's very prolonged immunity. There's been some very large real world studies in the VA system and other systems, and the level of protection that's reported in those studies is substantially below what was observed in the first clinical trials. There's somewhat controversial, but I believe it, data from very complete data sets from Israel that show that there's waning, including in a 6-month time frame for protection against both asymptomatic and symptomatic disease. And there's some controversy about how long you get protection, a very high level protection. And then the last thing is that, we talk about how long the protection is going to last, but is there a difference between the vaccine and the live virus exposure? And I'm not convinced that there's a difference. I'm not convinced that the protective effects of live virus infection really last for years and years, whereas the vaccines are waning. I guess the last thing I would say is that, in vaccinated individuals, there's a miraculous benefit. I can't count the number of lives saved, but there are large parts of the world which have simply not been vaccinated. And so the issue of longevity of immunity in the natural infection is unfortunately much more relevant in some parts of the world than the parts of the world which have been more heavily vaccinated.
I have a quick follow-up question, though, on what you were saying about the VA studies and the Israeli studies. I mean, in both of those cases, wasn't the average age quite high? So what you're talking about with the waning of the immunity, could that be very age specific?
Well, actually, I think the data is very good. Florian and others may have a different view, but the half-life of the antibody response across ages is approximately the same. It's 50, 60 days, including from natural infection and vaccine-mediated immunity. And I hear you that it may be that there's greater protection in younger populations, but there's also an aging population on Earth, and those people are the most vulnerable. So when I think about vaccine success, I'm more worried about the vaccine success in the most vulnerable parts of the population. So I agree that in younger ages, we may be talking about something quite different than the vulnerable populations. And the biggest risk is clearly age.
But speaking of a different group of vulnerable people, Dr. Barbouche, what about immunocompromised people? How should we be thinking about protecting them? I mean, are the vaccines really effective for them, or should we be focusing more on antiviral drugs and monoclonal antibodies?
Sure. Immunosuppressed patients and individuals are really an important target in this COVID pandemics. And I think that there has been a lot of work around these patients, and we know that people who are secondarily immunosuppressed individuals through treatment or transplantation, or are very susceptible to this infection and complication. So it's very important to vaccinate them. Yes, we should vaccinate them, and they should be vaccinated with specific protocols, because their immune system is not good enough to mount the necessary response like healthy individuals. But there are other immunosuppressed individuals in whom we are interested, particularly here in my country, which are primary immunodeficiency. And for some strange reason, these patients who have genetic defects in their immune system and should be particularly susceptible to this microorganism, like for others, have not been as severely infected and affected like what we were expecting. So this is another thing that we should look around. For example, people with combined immunodeficiencies, people with high gamma globulinemia, with common variable immunodeficiency, have not been as severely affected as we would have expected. High gamma globulinemia, I mean, they do not have immunoglobulins. So that's really very specific, and there's a lot of work which is performed now and done in this area. So yes, these individuals need vaccination, but they also need probably monoclonal antibodies for some of them, and when it's available, of course, and because this depends on the countries, and it's very important to say that with regard to my African continent, where those monoclonal antibodies are not always available, unfortunately, but then we also need to think about antiviral drugs, of course, which are not as advanced in terms of availability as we wish at this time.
Okay, yeah. Thank you for that. This is a sort of general question for anybody who wants to jump in, but we seem to be in a phase of the pandemic now where we are not so stressed out and in a crisis, and we can look back and think about what the best strategies are. Is there something that we should be doing differently in terms of the timing or the dosage of the vaccines, or mix and match strategies? Where do you think we should be going ahead?
Isn't the biggest lesson that even with miraculous, huge amounts of funding from certain governments and very rapid vaccine development and remarkable results, it's still too slow? And that what the real issue is that the vaccine should have been available for testing before the pandemic started, instead of reactively making the vaccine. I mean, I don't want to critique the vaccines. I'm happily vaccinated. I have that option. But there are a lot of people who could have benefited from a vaccine more rapidly available than we got, even with the remarkable timelines that we saw.
I mean, I want to comment on that too. This time I agree with Skip. I think in general, the pandemic preparedness is not where it should be, right? We were unprepared for this one. We did a great job getting the science done, getting the vaccine developed, but it's actually a sad state we're in, and I don't think we are better prepared for the next one. If this H5 that now infected one person in Colorado would take off, which I don't think it will, we wouldn't be any better prepared. And I think
I'm agreeing with you. As a species, we ignored the first warning, which was SARS, and then we got the second warning, which was MERS, and we ignored MERS, and and here we are. Florian's right. We are not much better prepared today for the next thing than, if at all, than we were for this.
Go ahead. So if I put you in charge, I'm in charge of the whole world, and I've put you in charge of the next generation of vaccines to be prepared. What are the kinds of vaccines that we should be looking at? What's the next gen look like? Should we be thinking about mucosal vaccines? Like, how would you be prepared?
So I think that would be relatively easy, but it would cost a lot of money, and a lot of money is relative, right? But I think what needs to happen is we need surveillance programs to figure out which viruses are out there. We need to have a better understanding of that. We know the virus families that can do things like that, and people are mostly worried about respiratory viruses just because they spread very quickly. viruses that need contact or a mosquito don't spread that quickly and can be easier stopped, right? But I think we need the surveillance. We need to understand the pathogenicity factors, and then you can select by doing phylogenetic analysis, and you can really drill in and select vaccine strains. And you can go to phase 1, you can go to phase 2 using different platforms. mRNA, of course, would be interesting, but also other platforms, and go into phase 2 trials, maybe larger ones where you start to understand immunity and safety. And once something hits, it's likely that that's actually related to what you prepared the vaccine for. If you prepared enough different vaccine candidates, and you can just switch and basically start a phase 3 trial and get that out quickly, maybe based on a correlate of protection. That needs a lot of preparation, it needs a lot of funding, and it also needs a lot of flexibility from the regulatory agencies. And I think that's also a main point. I'm still upset that it takes so long to make variant-specific vaccines and get them licensed. We are now expecting maybe that we might have some variant-specific or bivalent vaccines by fall. If a variant would come like Omicron, but would be much deadlier, which I hope will not be the case, and I think pre-existing immunity will attenuate that. But what would we do? We would have to wait for months until it's over, and then we would have a vaccine. And I think that's also really a regulatory hurdle. And I think the regulatory agencies need to think of how to do that quicker.
Emma, I have a question for you based on what we've been talking about. And so if we want to be prepared, what are the sort of top 5 viruses or virus variations that you are thinking about as where we should be keeping an eye, and what keeps you up at night? Like, which viruses do you think are going to come out of the blue and take us all by surprise?
I think the problem. Oh, go ahead, Emma.
Oh, sorry.
Yeah.
Sorry, was it for me or did I miss?
Yes, it was for you.
Okay, sorry, Florian. I definitely want to hear your thoughts on this as well, because it's a really tough question. I mean, I think that for one thing, I think is worth saying is that I think the chance that we predict exactly what will be the next virus that is pandemic is probably vanishingly small. And so one of the things that I really think about is broadening our understanding of viruses. We know, for example, coronaviruses, obviously they would be on our list now. Influenza's been on our watch list for a long time, possibly playing too much of a role. There's certainly hemorrhagic fever viruses out there that have been on our radar. But I think, what's the chance that it comes from one of these? What happens if it comes from families of viruses that we don't understand that well at all? And there's also a lot of viruses out there that may not be the most risky for something like the pandemic we've just had, but that are readily available around us. We have a lot of, the seasonal coronaviruses, enteroviruses, rhinoviruses that circulate around us all the time. And they live with us, they're endemic, we have immunity to them. And even these, there's a lot we don't understand as far as how they diversify, how much they cross-react, how long immunity lasts. I don't want to say there hasn't been work. There is some in it, and it's been wonderful. But there's still, so much we don't understand, even about the viruses that we get our whole lives. And this could teach us a lot about, how much cross-reactivity can we expect? How long will immunity last? How is that impacted over time through age with things like original antigenic sin? How much does it matter the first vaccine or the first infection you have? And how much does reinfection or re-exposure impact that? These are questions that we could be answering if we broadened our scope and invested in looking at a lot of different virus families, without needing to necessarily predict the next virus. And even if we still manage to not study the one family that it comes from, if we have a much broader understanding of all viruses, we'll still be able to narrow down better how this virus might respond, what our best plan of action is, how we should tailor our vaccines better than when we only have a really good understanding of 1 or 2 families.
That sounds, like a very smart strategy, but, in a very practical sense, when the governments are sort of allotting money, they do tend to prioritize certain things, right? So if you had to prioritize a certain vaccine, a virus family, I think coronaviruses, virologists knew that they were really dangerous, but a lot of other people just, as you said, thought about only the flu or maybe avian flu, but not so much thinking about all the other kinds. So what would you say is, an underappreciated, potentially dangerous family of viruses that we should be thinking about?
That's a tough question, and I'm not sure I have a really good answer for it, because certainly my expertise is not in studying these more kind of underappreciated viruses. So I'm not sure I'd be the one to answer the question. But I also think that, to some degree, we might need to have a little bit of pushback here for governments. It's true that funding agencies, this is what we've been interested in. And this is why, for example, has been a lot of investment into what to do if there's a flu pandemic. And that's really relevant. We want to be prepared for flu pandemics. They happen, we know that. But on the other hand, we also need to change that strategy a little bit and make it clear to funding agencies and governments that just pursuing the next most risky thing is not a very good preparation. We need to be broadening our understanding and our preparedness.
Dr. Barbouche, I want to ask, go ahead.
Yeah. May I just add one thing? I think that it's indeed difficult to say which family of viruses will be the next, but the most important thing, in my opinion, is to prepare the world, and not only some countries, but the world in such, the WHO is working on that, to prepare them to the next pandemic. And this will go through developing an alert and a response mechanism which will be efficient and where we will be collaborating all together, because it starts generally in remote areas, in countries where you do not believe that it could start there. So it's very important to have this kind of system that will be prepare us all as a whole, as a planet, to these kind of events.
Thank you for that. I was actually going to turn to you to ask exactly that question, because it doesn't seem like the next virus will come from countries that are already putting a lot of effort into surveillance. So, but to bring it back to sort of the main topic of discussion, which is vaccines too, in a lot of parts of the world, people have really just stopped caring about the vaccines. They're not taking the vaccines anymore, and vaccination rates have stalled. So what do we do about that? Not just now, but even in the future, to convince people of the importance of vaccination?
So is it a question to me?
Yes.
I can talk about my continent, Africa. I'm representing here Africa. I'm from the sub-region, North Africa, but I know the situation in Africa. And Africa has gone through this pandemic a little bit differently from what happened elsewhere. As you know, the African continent is a young continent, so maybe age has had to do with this. But despite the very low rate of vaccination, globally in Africa, with exception in a few countries, the community was infected, but not severely affected. We didn't see in our hospitals in many African countries, at least, this situation that has been observed in the States or in Europe with this pandemic. So I think, and we need to understand them both from the social point of view and from the epidemiological and immunological point of view. So vaccination has been, how to say it, encouraged in our countries, but there has been a lot of inequity of the vaccines in our countries. We started our vaccination campaigns with much delay compared to the Western countries. We used vaccines that were not already accepted by the WHO at one moment. So the situation was completely different in our countries. But I need to say that despite that, hesitancy was there. Yes, hesitancy was there. In my country, we have, despite all effort done, we have covered 60% of the target population, but with nothing to compare with European or US, of course. So we are still lagging behind in terms of vaccination, but how much is it important to these boosters? This is a real question today. The real hot question is about the boosters. What we should do, what we should recommend. The hesitancy is more important with regard to booster. In Tunisia, for example, in my country, 6 million out of 12 had got their first cycle of vaccination, but for the booster, only 1 million got the third dose. So the hesitancy is extremely important for the boosters. That's why we should be very, very careful about our strategies and decision with regard to booster. A fourth booster here doesn't make sense. Even if we are recommended it for particularly susceptible individuals, but in the general population, it won't work. So that's a real problem at the world level.
So that was actually a question I was going to get to later. The future. What does the future look like? How many boosters and how often and when in the year should we take them? So let's jump in now. I mean, what do you all think about how we should be thinking about boosters, not just in the near term, not just this fall or winter, but ongoing basis. Florian?
I mean, I can respond. I assume Skip has a different opinion. So I think, we have a system for influenza where the vaccine is given every year, and that might not be the smartest way to do it. We know that a lot of people who get the flu vaccine every year don't respond to it well anymore. They respond better if the intervals are longer. That doesn't mean it hurts. It's not making you more susceptible, but it might actually not give you optimal immunity, right? And so the question is if something like that would even make sense for SARS coronavirus 2. Now, the other question is, what is the virus going to do, right? And I think it depends a lot on that. It depends a lot on how the evolution looks like. If we see, again, a variant like Omicron that completely escapes existing neutralizing antibody responses, I think then boosters would be very important. And so what I imagine is a framework where two factors come into play. Waning immunity in some parts of the population, and then of course what the virus is doing and how the vaccine needs to be updated. So that's kind of how I think about it. But maybe somebody smart comes up with a vaccine that gives broader immunity, and maybe at least the virus evolution question is then for some time not really the problem. The other issue that I see is many of us have experienced that, specifically in North America and in Europe, the mRNA vaccines are used, and they do have relatively strong reactogenicity. A lot of people have to take a sick day after they get vaccinated, right? And so the question is, how accepted will that be? If you don't force people to do it, will they do it? Will they do it specifically if they know they are more or less facing a relatively mild disease if they get infected, which is now the case for young people with Omicron in many cases, right? So I think there's a lot of factors that come into play, and it's hard to imagine how this will work out in the future.
So I don't know whether it's going to surprise you, Florian. I agree 100% with every single thing that you said there. That was awesome.
All right.
I want to. So this issue of the yearly or X number of months, in the end, it's not really a good worldwide vaccine approach. Even with the flu vaccine, the relative uptake of yearly boosters across the world is wildly variable. And so what if you step back and say, what do we really want? So you want something that can prepare for variation, and you want it to last a lot longer than these vaccines currently last in efficacy, not just measurable immunity or immune responses. And to me, the thing comes down to antigen design. So Omicron evaded with 100 to 1,000-fold changes in neutralization by epitopes across almost all of the RBD, the receptor binding domain, and extensively within the RBM, the part that binds to ACE2, the receptor binding motif. But there were several clearly defined epitopes, and there have been a couple of others that were not altered. In other words, they're in common between the parental lineages, the Wuhan lineage, beta, delta, et cetera, and the Omicron lineage. I think it's a challenge that immunologists should take on to make vaccines that are variant proof. I think the structural biology, I think the capacity to map the epitopes, I think the biology of how antibodies work and how T cells work, what levels you need to maintain over a period of time is much more predictable than ever before. And I think we should spend the time, effort, and resources, including at the government level, to do that, so we don't have to get vaccines every year or every X months. And I feel like that sort of pan-virus or pan-Sarbeco or pan-influenza, Florian and others have made major contributions to thinking about this. That's what we should do. I mean, why can't we get to something that looks much more like the measles virus? And the answer is, well, you know, it evolves, it's evading immunity and everything. We can't change everything. They're conserved epitopes. T cell and B cell.
Emma, you seem like you had something to say to that, no? Okay. So, I mean, it's interesting because I've been hearing a lot about having a flu or flu plus COVID national campaign every year, and in your book saying that that's not a great idea. So I'm a little bit surprised by that. But also I've been wondering, how do you even get people to take it? Because at least in the US, when you have these flu campaigns, only about half of people sign up for it. So how do you actually protect populations? I mean, what do you think about vaccine mandates? Do they work? Like, what are ways that we can protect a population without either having a mandate or, you know, letting the virus run free?
Maybe I can add to something that Skip said. I mean, flu vaccines are basically vaccines that are used in high-income countries, right? Low-income countries either have other problems or they don't have the infrastructure and the budget to actually roll them out, right? And so I think even in the US and more so in Europe, the uptake for flu vaccines is low, right? So I think even if you would have an annual vaccine for SARS-CoV-2, it's questionable that a large proportion of the population would even take it. And I think in terms of the vaccine mandates, there was a lot of talk about that in Europe, and some European countries actually mandated, but then didn't implement, right? I'm originally from Austria. Austria mandated vaccines, but then they didn't implement the vaccine mandate, right? There is a strong opposition against that. And it's not just about mandating, it's also about, we have really a hard time to convince certain people to get vaccinated. I think that's one of the biggest challenges. Even with new viruses, and even if there would be a pandemic with a virus that has a 50% case fatality rate, you might have issues to convince people to get vaccinated. But that's not my field of study. I'm not a sociologist, I'm a virologist. So I have no answers for that.
I wanted to just kind of step in it very, very similar to what Florian is saying, and with the same caveat that I'm not a sociologist. And I do think that one of the biggest things we're going to have to overcome if we want to give more vaccines is to both make them much more globally available, but also to overcome some of this vaccine hesitancy. And I think one long-term thing we really have to think about there is better science communication and better education about how this works. I think unfortunately, well, maybe not unfortunately, but well, yes, unfortunately, but the mandates have led to a lot of backlash. I think they've kind of undone some of what could have been built up through more trust and more education and more realization of, you know, what the vaccine is, how they work, and what they aren't. But I also think, you know, that's a big thing, and that's going to be a hard thing to do, and it's not going to happen overnight. That's going to take long-term investment in how we communicate, you know, from school up through public communication of the government and science to the public. But there are also, at least in many countries, some easier things that we could overcome. Right now, in a lot of countries, vaccines are kind of divided into mostly two categories: the ones you have to get to start school, or that you're recommended to get, I guess having to start school is country variable. And then there are some that you're recommended to get, you know, for travel. And flu is kind of one of these ones that sits in the middle, that it's like recommended, but everyone should get it, and it's not travel dependent. And we've never, in a lot of countries at least, have never invested much into caring much about how many people are getting it. And in a lot of countries, we still have really basic barriers like cost. Here in Switzerland, it's like 40 francs, that's $40 US to get the flu shot. Well, that's a lot of money for, you know, a virus that a lot of people think I'm not going to get anyway. In other countries in Europe, you can't get a flu shot without making an appointment at a doctor's office. Well, that's a barrier that's going to turn loads of people off, even if you don't have to pay to see that doctor. It's an appointment and time and everything else. So I think that we should maybe also just be thinking about, if we are in a world in the future, which I think is not impossible, where vaccines are more readily available for more things, and we want people to get them more often in their life, how do we make that more appealing to people, even in really basic ways? And those are things that I think are not easy to change, but they're also not impossible to see how we could start doing that.
May I add one thing very rapidly? Yeah.
Go ahead.
Because I'm Rita. As an immunologist, I believe that vaccines are useful and are needed really, and still for COVID, we will need to have good vaccines. And after all the work that has been done in the last months and years, I think that a good mucosal vaccine, for example, with this availability of good level of IgA, of memory, T and B cells locally, would be a fantastic thing and could help solve part of the issue we are confronted to. I think that to identify universal vaccine against all coronaviruses would be also a good advance. So there is still room for a better vaccines than what we have today. And this will also depend on the virus itself, of course, and the evolution as we have discussed. Thank you.
Thank you so much. This has been a really fascinating conversation, and I thank you all for letting me put you on the spot sometimes and pushing you maybe further than you're comfortable. I actually, unfortunately, have to go because I have another thing, but if the conversation is going to continue on, I want to hand off to Faith, and maybe she can either figure out what to do. But thank you all so much. This has been a great pleasure.
Thank you, Apoorva, and thank you, panelists. It's been really, really nice. Skip, I'm sorry I missed you at the introduction, but Federica jumped right in and sorted us out. I think there's such topical issues that came up in the discussion that we're all thinking about. And so thank you for sharing your thoughts. It's been a fantastic time, and there's a lot ahead of us. You know, we hoped we'd be further ahead at this point in the pandemic, but really there's a lot to go. And I think a lot of fundamental immunology that's also still required. So I want to just thank you all so much. Thanks to the panel. Thank you for your time. Thanks to Apoorva. You know, we really appreciate you. Thanks to all our attendees from all over the world for staying long. You know, it's been a good 3 hours. Thank you to our organizers, to KIT, to the IUIS for bringing the event together, and EFIS. Federica, thank you. I think we can say goodbye.
Goodbye.